Okay, in this first one about the genetics. What is the meaning of the genetics? Genetics, that means it is the science. It is the science of heredity and hereditary variations in living organisms. It is the branch of the biological science and study about the heredity and hereditary variations in living organisms. That is called genetics. Genetics is the term coined by William Bateson in 1905. Okay, student. Next, student, what is the meaning of heredity? Heredity, that means study of transmission of characters from one generation to the next generations. Student, different types of the characters: physiological characters, physical characters, morphological characters, structural characters. These are the characters which are present in the one generation or the parents and inherited or transmitted to the next generation. Okay, that is called heredity, transmission of the character. About the study of the transmission of characters, that is called heredity. Okay, student, these are characters which are transmission from the one generation to the next generation, called hereditary characters. Next, variations. What about the variations? Variations means differences in characteristics shown by individuals of species, also by progeny of some parents. And in this, variations means differences in characteristics. Differences are available in the species individuals of the single species also shows the much amount of the variations. Variations are highly available in different species individuals, different phylum individuals. Okay, student, these are the variations. Variations means differences in characteristics, size, shape, color. These are body structural organization and body parts arrangement and shape. It's a shape. These are the different types of the characteristic also available in individuals belongs from the single species. This is called variations. Differences in characteristics shown by individuals of species, also by the progeny of some parents, progeny of same parents. Individuals that are came from the same parents also shows the variations. Student, inheritance. Inheritance, that means a process by which characters pass from one generation to the next. Students, characters, hereditary characters, heredity and variations belongs from the one generation to the next, passes from one generation to the next generation. Amount of the process involved in this passing of characters, that is called inheritance. Passing of the characters from the parents to the next generation, parents to the generals, parents to the progeny, parents to the children. In this, which amount of the process is involved in it, that is called inheritance. So it is called inheritance of the characters from one generation to the next. Next, student, Gregor Mendel, considered as the father of genetics. In modern science of the genetic, to understand the involvement of the process of inheritance, it is begin with the work of the Mendel. Process of the inheritance, which is involved in the modern science of the genetics, is initiated and only begin with the work of the Mendel in mid-19th century, in the middle of the 19th century. Student, Gregor Mendel known about the passing of the inheritance characters from one generation to the next generation on the observation on the pea plant. Okay, but he did not know about the which amount of the physical basis, physical factor involved in it. Okay, these are Gregor Mendel, the passing of the characters, passing of the inherited characters from one generation to the next generation. It is possible by the some amount of the inherited units. Okay, but he did not know about the which type of the factor involved in transmission of the inherited characters. Mendel's work was rediscovered in year 1900 by three scientists: Hugo de Vries from the Netherlands, Correns from Germany, Scherman from Austria. Three scientists tried to determine which amount of the unit involved in the transmission of the characters, transmission of the hereditary characters. Okay, these three scientists determined one of the molecule present in the cells of the body, cells of the organism. These are responsible for the transmission of the inherited characters from one generation to the next generation. Okay, Hugo de Vries, Correns, Scherman. Next, under Sutton-Boveri theory. Sutton-Boveri theory. What about Sutton-Boveri theory? This is also called chromosomal theory of inheritance. Okay, Gregor John Mendel determines and discovers one of the factor involved in the transmission of the characters and called as units of inheritance. And Hugo de Vries Correns Scherman, after his rediscoveries of Mendel, and determines molecules present in the cells. These are responsible for transmission of the characters. And Sutton-Boveri theory proposes chromosome theory of inheritance. Identify chromosomes as carriers of genetic material, basis for genetic inheritance. Okay, Sutton-Boveri, two scientists, conclusion with chromosomes are responsible. Chromosomes present in the cell, these are responsible for inheritance of the characters in the generation to next generation. Question: these chromosomes are considered as genetic material, and these are carriers to transmit the inherited characters, and it is the basis for genetic inheritance. Sutton-Boveri theory, also called chromosomal theory of inheritance. Next, student, under Thomas Hunter Morgan, T.H. Morgan, Thomas Hunter Morgan, chromosomal theory of inheritance experimentally proved by Thomas Hunter Morgan. Okay, for his experiments, Thomas Hunter Morgan selects on his observation, on his experiments, selects one of the animal, that is a fruit fly. Okay, fruit fly, selected fruit fly. Scientific name is Drosophila melanogaster. Fruit fly scientific name is Drosophila melanogaster. Okay, student, why he selected Drosophila melanogaster fruit fly? Because some amount of the easily observed characters are present in this organism in this animal. Okay, student. Drosophila melanogaster. Okay, which are the characters, easily observable characters are present in Drosophila melanogaster? This causes for the selection of this animal for his experiments. Okay, chromosomal inheritance theory experimentally proved by T.H. Morgan. Okay, Drosophila melanogaster selected for, which are the causes for the selection of the Drosophila melanogaster? In this first one, grown on simple synthetic medium in the laboratory conditions. Okay, this is first one, and complete its life cycle in about 2 weeks. Entire life period of Drosophila is completed in 2 weeks only, and single mating produce a large number of the progeny. Single mating between male and the female, that produces a large amount of the next generation or the progeny. Okay, next and the student, fourth one, many types of the morphological hereditary variations can be seen under low power microscope. Student, this is the small animal Drosophila melanogaster and shows much amount of the morphological characters. Morphological characters means characters that appeared on the body surface, body parts, body arrangement externally, and size, shape of the body, color of the body. These are morphological characters. Much amount of the morphological characters and hereditary variations also much. These are all the characters also easily observable under low power microscope. Next student, fifth one, has only 4 pairs of chromosomes. It is also one of the easily observable characters. Only 4 pairs of the chromosomes present in these animals. Okay, if you observe cells of the body of Drosophila in the nucleus, that total number of the chromosomes is 4 pairs, only 8 in number. 4 pairs of chromosomes, and these are easily differentiation of these chromosomes, distinguishable of these chromosomes are easily under light microscope. Okay, the 4 pairs of the chromosomes Drosophila melanogaster, 3 pairs are autosomes. 3 pairs are autosomes. Okay, 6 chromosomes are autosomes, and 1 pair 6 chromosomes. 6 chromosomes are 1 pair. In this, student, if you observe female fruitlets, 1 pair homologous X chromosomes. Homologous X chromosomes, 1 pair. Okay, these are 6 chromosomes. 6 chromosomes, 1 pair. If you observe female Drosophila, these are 1 pair with X chromosomes. Next, if you observe male one that consists of 1 X chromosome and another 1 Y chromosome. That is X and Y chromosomes. Okay. In female, this is the 1 pair, 6 chromosomes, and if you observe male one, XY, this is the 1 pair, 6 chromosomes, and remaining are autosomes, 3 pairs of autosomes. These are same in females and males. Hi students, I'll discuss about concept of linkage by Morgan experiment. Already you know about the chromosomal theory of inheritance. According to chromosomal theory of inheritance, inherited characters are inherited from the parents to the next generation by passing of the molecules which are present in the form of chromosomes in the cell. Okay, these are carries the same type of the information from one generation to next generations. Okay, this is the chromosomal theory of inheritance experimentally proved by Thomas Hunt Morgan. For his experiment, T.H. Morgan selected one of the animal, that one is Drosophila melanogaster. It is the scientific name of a fruit fly. Okay, Morgan goes to dihybrid cross on Drosophila. What is the meaning of dihybrid cross? That means mating between two individuals, that is cross. Cross means mating between two individuals. And dihybrid, that means with considering two pairs of the contrasting or distinguishable characters, that is dihybrid cross. Next student, Morgan goes to dihybrid cross and Drosophila melanogaster, okay, to study about the independent inheritance of the two pairs of characters. Morgan observes Drosophila for passing of the inheriting of the two types of characters. Okay, two pairs of the characters in single animal. Okay, next student, and he wants to know gene for body color. One is the character is body color, one is the character. Two types of the characters he observed in Drosophila at the time of the experiment. Okay, one is the body color, second one is wing size. Okay, this is Drosophila is insect, belongs from the phylum Arthropoda. Okay, were genetically linked. These are the characters, phenotypic characters such as body color, wing size are shown by the animal because of responsible genes present in the karyotype. Okay, these are genes or genetically are linked up. And what about this effect of their inheritance, students? Next student, one gene with two alleles. Already you know, one gene available in the form of the two allele. Okay, allele of body color gray, it is the phenotypic character, one of the body color in Drosophila, gray color. Okay, allele for body color gray, B plus. Okay, next allele of body color black, that is allele is B. Okay, only B. Next student, allele of wing size long, that is VG plus. Okay, allele of wings vestigial wings, that allele is VG. Okay, so VG means vestigial. Students, and the Morgan selects two parents, okay, in Drosophila, genetically different parents. One is male and the one is female. Long wing gray body, long wing, that is the gene with two alleles. One is VG plus and the one also VG plus, because of these are pure animals. Two alleles are same because the animal that shows the long wing, okay, alleles are VG plus and VG plus. Gray body alleles are B plus. Okay student, according to this. Next, vestigial wing VG and black body small b, only B. Okay, and these are the genes present in mother cell which are goes to the production of the gametes If you observe the gametes, half of the allele entered into the gametes. Okay, Vg plus B plus, only one of this one. Okay, Vg plus B plus, Vg and B. Okay, these are the gametes. Gametes are fused with each other and form the zygote. Gametes goes to fertilization and forms the zygote. Zygote develops into young one. Okay, growth into adults, and these are called first generation, F1 generation. F1 generation is with both are combined, Vg plus Vg, B plus B. These are the progeny having long wing and grey body fleece. Okay, student, these are males or females. Next, student, another again mated wild type F1 hybrid female. In these animals, Morgan selected female ones having long wing and grey body fleece. These are F1 hybrid female and mated with black vestigial winged male. Okay, student, and offsprings are produced. F2 generation is also produces. These are all the organisms, fruit fly, Drosophila animals having parental phenotypes. Which are the phenotypic characters such as grey body, black body and long winged vestigial wing? These are only characters are appeared in the F2 generation. All are the phenotypic characters are present in the F2 generation. These are shown by the parents. A student, Morgan finally concluded with genes for body color and wing size are genetically linked on same of the chromosome, on the same chromosome. Okay, student, this is about the concept of linkage, Morgan experiment. Students, it is the topic about pleiotrophy. Pleiotrophy. What is the meaning of the pleiotrophy and which one is the example for pleiotrophy? In this, for example, students, specific gene, if you observe one of the gene controls specific phenotype. So many types of the phenotypic characters that is shows by the organisms. Okay, every one of the characters present in the animal, that is by the presence of the, that is caused by the presence of the genes in the karyotype of the animal. Okay, each gene, specific gene controls specific phenotype. Phenotype means character, showing character by the animal. Student, it is not always true. After the studies on the gene expression of the genes, after the studies on this, it is not always true. Okay, which one? Specific gene controls specific phenotype. Okay, single gene, according to this, a single gene influence more than one phenotypic trait. Okay, single gene that causes for the appearance of the multiple phenotypic characters. Okay, more than one phenotypic trait. Okay, students, in this multiple effects of single gene. Okay, one of the gene responsible for the appearance of the more phenotypic character, that is called pleiotrophy. Student, pleiotrophy. Okay, it is important, students. In this, in human beings, if you observe in human beings, the best example for pleiotrophy is one genetical disorder, that is PKU, that means phenylketonuria. Phenylketonuria is the best example for pleiotrophy. Student, if you observe amino acid phenylalanine, what is the metabolism of the amino acid phenylalanine? Student, in this phenylalanine converts into tyrosine by the phenylalanine hydroxylase enzyme. It is the hepatic enzyme, liver enzyme. Phenylalanine hydroxylase converts the phenylalanine present in the food, one of the amino acid phenylalanine converts into tyrosine. Okay student, what about the phenylalanine hydroxylase? That is the responsible gene. This is the synthesis of this enzyme is by the one of the gene that goes to the expression that codes for the synthesis of this enzyme. Which one is the gene? That is the gene locates on the chromosome 12. If you observe karyotype of the human beings, entire total 46 chromosomes are there. Okay, in diploid cell 46 chromosomes, 23 pair chromosomes are present. In this 12th chromosome, where it is present, on the 12th chromosome, one of the gene is locates, that is the gene synthesize hepatic enzyme called phenylalanine hydroxylase. Okay, which one is gene that codes the pH? That is the gene goes to the mutation, that is goes to some amount of the changes. Okay, these are the changes are inherited to the next generation. Okay student, that is the mutant gene inherited to the progeny, next generation from the parents. So it is the phenylketonuria is the genetic disorder student, that is the mutant gene not synthesize phenylalanine hydroxylase. Okay student, in this phenylalanine hydroxylase is not produced because of mutated gene. Okay, phenylalanine at that conditions, phenylalanine is converts into phenylpyruvic acid because of absence of the phenylalanine hydroxylase. Okay student, this is the phenylpyruvic acid accumulates in the blood, increases the level of the phenylpyruvic acid in the body fluids, that causes the appearance of the multiple phenotypic characters, multiple phenotypic traits in the human individuals. Student, cause multiple phenotypes with the phenylketonuria includes mental retardation. Mental retardation means it decreases the level of the thinking. Okay, next reduced hair and pigmentation. Pigmentation, that means coloring, changes the coloring of the body. Okay, by the presentation of the level of pigments. Okay student, these are the multiple phenotypic characters are they shown by the genetical disorder, that is phenylketonuria. Students, now discuss the topic is multiple alleles, human blood groups. Students, firstly, one gene with two alleles, one gene having two forms, these are called alleles. One gene with two alleles, okay, present on same locus of pair of homologous chromosomes. Student, homologous chromosomes means these are similar. These are similar type of the chromosomes, same type of the chromosomes. Okay student, on the locus of homologous chromosomes, two alleles are present. Okay, one of the gene is present in the form of two alleles. These alleles are present on locus of homologous chromosomes. Okay, one gene with two alleles, for example, one gene with two alleles, okay, that having three genotypes, okay, because of presentation of the two alleles, that causes the appearance of the presentation of the three genotypes in diploid organisms. Diploid organisms means the cells are diploid, having two sets of the chromosomes in the cell. Okay, if you observe the karyotype of this cell, karyotype means the chromosome complement of this cell is called karyotype. Okay, student. Another, one gene more than two alleles, one gene more than two alleles, student, that is called multiple alleles. One gene more than two alleles, okay, normally one gene with two alleles, but another conditions, one of the gene more than two alleles, that is called multiple alleles. It is the phenomenon present in the population. It is the phenomenon, that is presentation of the multiple alleles is called multiple allelism. Student, for example, capital T, small t, or two alleles for one gene. Okay, these are the two alleles, capital T, small t, are the alleles for one gene. One gene with two alleles, because of one is the small t, one is another one is the capital T. Okay. Next, genotype, if you observe, capital T and capital T, small t, and small t, these are the genotypes. One gene with two alleles, and at the same time it is there is a possibility of the three genotypes. These are the alleles, these are the genotypes, three genotypes. Okay, student. Okay, these are, by the presentation of these alleles, the organism shows the different types of the phenotypic characters, morphological characters based on these. Okay, this cannot observe, this is the multiple allelism, cannot observe in the genotype of the individuals. Observation is possible only within the population. Okay, you have the multiple alleles, number of the multiple alleles, and by the number of the multiple alleles you also obtain or estimates the number of the genotypes. Okay, by the expression of the, by the formula of the n into n plus 1 by 2. Okay, n into n plus 1 by 2. Okay, n is equal to number of the multiple alleles, number of the multiple alleles. For example, students, multiple alleles or alleles are 2 in number, alleles are 2 in number, n is equal to 2. Okay, student, number of the multiple alleles, n is equal to number of the multiple alleles. Okay, it is written with 2 into 2 plus 1 by 2, that is 2 into 3 by 2, 6 by 2. Okay, student, answer is 3. The genotypes are 3 in number. Okay, 3 is the genotypes. Okay, if the alleles, number of the alleles are 2, how many genotypes are present? 3 genotypes are present. Okay, next, ABO blood types. ABO blood types. Number of the multiple alleles, if you know, and number of genotypes is estimated by the formula n into n plus 1 by 2. Okay, student, n is replaces with the number of multiple alleles. Next, which one is the best example to studying multiple allelism or multiple alleles? ABO blood types, proposed by Carl and Steiner. ABO blood groups, AB, A blood group, B blood group, AB blood group, O blood group, these are proposed by Landsteiner. Okay, and because of his observation, Carl Landsteiner is also awarded with Nobel Prize, okay, in physiology or the medicine in 1930. In the year of 1930, Carl Landsteiner is also awarded with Nobel Prize in medicine. Okay, A, B, O, these are the blood types. These are the blood types also called phenotypes. Phenotype means that is the character for blood group. Okay, phenotype it is also called. It is the one of the character by the gene. Okay, next by the presence of absence of the antigens on membrane of a red blood cell. Student, okay, this is the phenotypic character A blood group, B blood group, based on the presence of the antigen on the membrane of red blood cell. Student, if you observe the red blood cell, membrane of the red blood cell, membrane, this is the membrane of the red blood cell. Okay, that is shows the antigens. These are the antigens, students. These are the antigens present on the membrane of RBC. Which type of the antigen present on the RBC that gives the different types of the blood group? Okay, antigen A, antigen B. These are the antigens are two types present on the membrane of red blood cell. Okay, antigen A one type, antigen B another type, two antigens, antigen A, antigen B on the RBC, plasma membrane of RBC. Student, what about these antigens? These are blood group antigens only. Okay, carbohydrate groups, antigens are carbohydrate groups or sugar polymers. These are the sugar polymers, carbohydrate groups or sugar polymers, and bond to the lipid molecules, present on the membrane of RBC and slightly extend to the outside from the membrane of the RBC, surface of the RBC cell. Okay, student. Okay, slightly exposed to outside. Okay, present on the surface of the red blood cell. These are the antigens. These are carbohydrate groups or sugar polymers. Okay, these are the antigens also called isoagglutinogen. Antigens, blood group antigens present on RBC also called isoagglutinogen. Why these are called isoagglutinogen? Because blood cell agglutination, blood cells present in the body fluids, blood goes to agglutination. Okay, student, in case of incompatible blood transfusions, in case of incompatible transfusion of the blood, these are the blood cells goes to the agglutination or the clot. Okay, student, these are called isoagglutinogen. Causes agglutination. These are the antigens causes agglutination in case of incompatible transfusion of the blood. Hence, these are the antigen be called isoagglutinogen. Okay. Next, Bernstein. What about the role of the Bernstein in study about the ABO blood groups in human blood groups? Okay, which are the best example for multiple allelism? Bernstein, another scientist, phenotypes inherit by interact of three autosomal alleles of gene I. Okay, student, blood groups present in the individuals of human beings. This is the one of the character, phenotypic character. Individual shows A blood group, B blood group, AB blood group, O blood group. This is one of the phenotypic character by the presentation of the gene. That is, the gene is I. I is the gene, student. I is the gene. Okay, and this is the gene with three alleles. This is the gene with 3 three autosomal alleles. Autosomal alleles. Chromosome is the autosomal chromosome. That is the gene locates on the locus, present on the locus of the ninth chromosome. Ninth chromosome. Ninth chromosome is the autosome. It is not a allosome or sex chromosome. Student, chromosome is ninth chromosome. Gene is I. I is the gene with three alleles. Okay, three alleles. Okay, because of presentation of the three alleles, how many genotypes are produces or obtained by the formula of n into n plus 1 by 2? Student, in this 6 genotypes are obtained. 6 genotypes are obtained. Okay, antibodies. Now what about the antibodies? Antibodies, that means these are antibodies are two types. These are students blood group antibodies. These are blood group antibodies. Antibodies are present in plasma. This is very important. Antigens present on the plasma membrane of red blood cell. Antibodies present in the plasma of blood. These are two types: anti-A, anti-B antibodies. Anti-A antibody, anti-B antibody. These are called isoagglutinins. These are called isoagglutinins. Antigens are called isoagglutinogens. Okay, student. These are called isoagglutinogens, antigens. Antibodies called isoagglutinins. Okay, these are IgM type of antibodies. Antibodies are different type. From this, these are the antibodies. The blood group antibodies are IgM type based on the structure. Next, cause agglutination. Why these are called isoagglutinins? Cause agglutination reaction. These are the antibodies cause agglutination reaction in the blood. Okay, with antigen of another individual. Antigen of another individual. Okay, student, these are the antibodies. Antibodies reacts with the antigens, okay, of the another individual and causes the agglutination, because called isoagglutinins. The gene is the gene with three alleles. Three alleles, student: IA, IB, IO. Okay, IO also called I. Student, IA allele, it is causes the production of the antigen A. Antigen A. Students, antigens are two types. Antigens are two types: antigen A, antigen B. IA, it is causes the production of the antigen A. IB, it is causes the production of the antigen B. Okay, IO, this is not causes the production of the antigens. Okay, IO, there is no antigens. There is no antigens. Okay, student, it is not responsible for the production of the any type of antigens. No antigens. IO, no antigens. Okay, students, IAIB, child receive IA from one parent, IB from another parent. Okay, student, IAIB. Student, these are dominant to IO. Okay, student, for example, one of the child receives IA from the mother parent, and this is the allele from the mother parent, and another one is from the father. In these two alleles, which one of the blood group is shows by the individual or the child in these two types of the characters, two types of the alleles. Student, at the same time, the individual shows the A blood group. A blood group. Okay. A blood group. Okay, student. Because IA is the dominant to IO. Okay, student. IA is the dominant to IO. Because that is the child is shows the A blood group. Okay, same student, IA, IB dominant to IO and co-dominant to each other. Student, at the same time, IA is from the mother, IB is from the father. Okay, child receives this type of the alleles from the parents. IA is the one, IB is the one. At the same time, this child shows the AB blood group. AB blood group. Okay, because co-dominant to each other. Co-dominant to each other. Student, AB blood group is present in that individuals. IA, IB dominant to IO and co-dominant to each other. Student, child receive one allele from the each parent give rise to 6 possible genotypes and 4 phenotypes. 4 phenotypes. Student, already you know, I is the gene, 3 alleles, IB, IA, IB, IO. By the presentation of the 3 alleles, 6 genotypes are produces. 6 possible genotypes. Gives 6 possible genotypes. Which are the genotypes? One is the IA and IA and IB and IBI, IA and IO. Okay, these are 6 possible genotypes. And how many phenotypes? And at the same time gives rise to 4 phenotypes. Which are the phenotypes? Which type of the blood group shows by the individual, that is called phenotypes. 4 types of the possible phenotypes. One is the A blood group, another one is B blood group, and the AB blood group, and the O blood group. These are the 4 possible phenotypes. Remaining part of the ABO blood groups. Student, it is the table about the genetic control of human ABO, AB blood groups. Okay. This first one genotype, second one antigens present on RBC, and ABO blood group phenotypes and antibodies present in plasma. Next, blood types that can be allowed into the individuals or the persons. Blood types that can accept the blood for the transfusion. Okay, student, in this first one, genotype, these are the genotypes. Okay, I is the gene with the 3 alleles. Okay, IA, IB and capital I. These are the alleles. Presentation of the 3 alleles and possibility of the upcoming 6 genotypes. Okay, these are the genotypes, different types of the genotypes. Receives from the parents to the next generation individuals. Individuals having 3 these type of the genotypes. Okay, first one, student, if you observe genotype IA, okay, and blood group is type A. Okay, blood group is type A. Okay, if you observe type A blood group individuals and which one of the antigen present on the RBC, that is A, antigen A. Next, antibodies present in blood plasma. Okay, student, anti-B in the plasma of the A blood group persons having anti-B antibodies. Okay, Okay, student, blood type that can be allowed. Student, these are the persons allows the blood of the A blood group persons and O blood group individuals. Okay. Next, blood type that can accept blood for transfusion, that means, student, these are the type A blood group individuals allows the transfusion of the blood to A group individuals and AB group individuals, AB blood group individuals. Okay, student, these are the different types of the phenotypes according to the blood group of the individuals, and these are the types of antigens on the RBC. Okay, these are the genotypes of the individuals. Okay, and antibodies, these are the antibodies, different types of the antibodies present in various types of the blood group individuals. Okay, student, it is the table about the genetic control of the ABO blood groups in the human beings. Students, blood typing, that means A blood group, B blood group, AB blood group, O blood group. These are the blood groups of the individuals can be determined by adding of the anti-serum which is present in the laboratories. Okay, and these are also called anti-A antibodies, anti-B antibodies. These are the antibodies are mixed with the sample of the individual blood and also can determine at the same time can determine the blood group of the individuals. Student, anti-A antibodies, anti-B antibodies. These two types of the antibodies also called anti-serum present in the laboratories. Students, when clump with the anti-A serum, individual's blood is mixed with the anti-A serum and clump is forms. Clump means group. Student, bunch like structure. Okay, group like structure, clot like arrangement. Clump with anti-A serum, that is the blood is A group or AB blood group. Okay, and clump with anti-B serum, when anti-B antibodies added to the individual's blood, sample of individual's blood, and clump is forms, that type of the blood is B blood group or AB blood group. Next, student, clump with both of anti-serums. Clump is formed with anti-A antibodies, anti-B antibodies. That individual's blood is AB blood group. AB blood group. No clump with either serum. When blood is mixed with the anti-A serum and anti-B serum, anti-A antibodies, anti-B antibodies, but no clump is available. Clump is not formed. Okay, that individual's blood is O blood group. Okay, student, in this way individual's blood's groups are determined. Students, now about blood transfusion. While transfusion of the blood from the one individual to the another individual, which type of the antigens present in the donors? Two persons are there, two individuals are there. One is the donor, okay, and another one is the recipient. Donor, recipient. Blood is transfused from the donor to the receivers. And which type of the antigens present about the blood group? Which type of the antigens present in the blood of the donor? And which type of the antibodies present in the receiver? That can be taken into consideration mainly at the time of the transfusion of blood. It is important. Okay. Which type of the antibodies present in the donor? Which type of the antigens present in the receiver? that cannot be taken into the consideration. Okay. In this, universal donors, which are universal donors? Persons with O blood group are universal donors. Why O blood group individuals are called universal donors? Student, if you observe O blood group individuals is with the absence of the antigen A, antigen B. Okay, student, both of the antigens are absent in O blood group individuals. Next, another, because of absence of the antigens in O blood group, these are individuals. Student, individuals are act as universal donors. O blood group persons act as universal donors because agglutination does not occurs with any other group of blood. There is no chance for agglutination because of absence of the antigens. Next, universal recipient. In this universal donors, by receiving of the O blood group persons, antigens are absent in that type of the blood. Antigens are not entered into the receiver's blood. That is important. Okay. Next, universal recipient. AB blood group individuals are act as universal recipient. Okay, student. AB blood group. These are the individuals are universal recipient. If you observe these individuals in blood, anti-A, anti-B antibodies are absent. Anti-A, anti-B antibodies are absent. Okay, by the absence of the antibodies, individuals does not shows the opposite action against the antigens. Any antigens enters into the receiver's blood such as AB blood group persons. Student, because of absence of the antibodies, agglutination reaction does not occurs. Okay, student, that is about the universal recipient. Okay, AB blood group persons, if you observe plasma, no antagonistic antibodies. Okay, and this does not cause agglutination of RBC received from persons of other blood group individuals. Friends, now discuss about RH blood types. RH blood types. Already you know about the different two types of the antigens on the blood groups. Antigen A, antigen B. These are present on the plasma membrane of red blood cell. Okay, and two types of the antibodies also there. Anti-A antibodies, anti-B antibodies. These are the antibodies for the blood group present in the plasma of the individuals. Okay, student, if you observe A blood group persons, type A blood group, in these individuals, antigen A present on the plasma membrane of RBC. Okay, and if you observe plasma of that individual, anti-B antibodies present in the plasma. If you observe AB blood group person, antigen A, antigen B, two types of the antigens present on the plasma membrane of RBC, and at the same time antibodies are absent. Okay, student, like this. Okay, next, and these are the antigens, presence of the antigens on the RBC. Antigen A, antigen B present on the plasma membrane of RBC. It is the phenotypic character that causes the A blood group, B blood group, AB blood group and the O blood group. Okay. By receiving of the different types of the genotypes from the parent generations. Okay. Inheriting of the genes in the form of the alleles about the phenotypic character that shows by the individual is receiving from the parents to the next generation, that causes the appearance of the different types of the phenotypic characters, different types of the blood groups. Next, students, now going to discuss about the RH blood types. RH blood types. Okay, already you know about two types of the antigens about the blood group. One is antigen A and the one is antigen B. In this, Karl Landsteiner, okay, and Wiener, two scientists discovered another one of the antigen, that is RH antigen, also called RH factor. This is also present on the RBC, plasma membrane of RBC, red blood cell. Student, it is the RH antigen also present on the plasma membrane of RBC, like antigen A, antigen B. Okay, student, these are firstly discovered by Landsteiner and Wiener on RBC of rhesus monkeys firstly, another later also discovered in human beings. Okay. Additionally, one of the antigen is also present on the plasma membrane of RBC, that is RH antigen, also called RH factor. Okay, RH factor, that is also called D antigen. D antigen. Okay, that are about the blood group antigens, that are antigen A, antigen B. This is the RH factor is also called D antigen. Okay, student, you can identify. Next, person with D antigen. Which are the person? All of the person does not shows the RH antigen on the RBC. Some amount of the persons having RH antigen. Which are the persons with D antigen on plasma membrane of RBC called RHD positive or RH positive? Which are the persons having D antigen? That are individuals are called RHD plus, RHD positive. Okay, and RH also called RH positive. Without the D antigen, RH antigen, RH factor is absent on the plasma membrane of RBC in some individuals. That are the persons are called RHD negative or RH negative. RH negative. Okay, students, and RH negative person, this is the D antigen is absent in that persons. RH negative person receives RH positive blood cells at the time of the donation. RH negative person receives RH positive blood cells, RH positive blood cells from the another one of the individual. Okay, first time anti-D antibodies are produced. Okay, immune system identify that entry of the RH positive blood cells by showing of the RHD antigen. Student, D antigen is present on the plasma membrane of RBC, that is the antigen. Okay, that is the new antigen enters into the body of the RH negative person. Immune system this identified and produces certain type of the specific type of the antibodies. These are called anti-D antibodies. Anti-D antibodies. Okay, student. These are the anti-D antibodies are normally absent in the plasma of individual. Okay, only these are produces at this time only. Okay, next Rh positive person receives Rh negative blood cells. Rh positive person, it is the person is with Rh positive having Rh antigen. Okay, receives Rh negative blood cells. Rh negative means this is the antigen is absent. Antigen is absent because there is no problem because of absence of the antigens. Okay, without any complications are there. Okay, student, it is about the Rh factor. Next, what about the genetic control of the Rh factor? Okay, students, in this, which one is the reason for the presence of this phenotype in the individuals? Which are the genes, which are the alleles are responsible for the showing of the phenotypic character, that is D antigen, Rh antigen, Rh factor. Okay, student, it is given by one of the hypotheses about the genetic control for Rh antigen, that is Fisher-Ray's hypothesis. Fisher-Ray's are two scientists give one of the hypotheses about the Rh factor, genetic control of Rh factor, that is Fisher-Ray's hypothesis, also called CDE nomenclature. What about the CDE? CDE nomenclature, it is also called. Student, in this, according to this hypothesis, allele for Rh factor, allele for Rh factor, student, presence of the D antigen, presence of Rh antigen, okay, that is the one of the phenotype is caused by the presence of the gene and their alleles. Okay, student, alleles for Rh factor, alleles for Rh factor, student, occur in form of the three pair. These are the alleles are available in three pair, in form of three pair at three different loci on chromosome pair 1. Student, if you observe chromosome complement of the one individual, one of the person, total 46 chromosomes are there, that means 23 pairs. In this 23 pairs, first pair, if you observe first pair of the chromosomes, these are homologous chromosomes, one pair, first pair chromosomes having loci. In that loci, three pair of the alleles are present. Three pairs of alleles are present. Three pair at a different loci on chromosome pair 1. Student, okay, Rh factor by three pair of Rh alleles. Rh factor, presence of the Rh factor, showing of the phenotypic character like Rh antigen, Rh factor by the three pair of Rh alleles. Three pairs of Rh alleles. Students, and under three pairs of Rh alleles, which are the three pairs of Rh alleles? Three pairs of Rh alleles belongs from the information on the one pair chromosomes. Okay, these are capital C small c, capital D small d, capital E small e. These are three pairs of alleles present on the different loci, three different loci on chromosome pair 1. It is the one chromosome, it is the another one of the chromosome. One pair chromosomes, first pair of the chromosomes these are, and this indicates the different loci. It is the one of the loci, another one of the loci, three different locis. Okay, student, these are three different loci. okay, on first pair chromosome. Student, capital C is the allele, small c is the allele, and capital D or small d, capital E, small e. Okay, in these two, capital C, small c, in these two only one allele is present. Okay, in this capital D, small d, it is the one of the chromosome, first chromosome. These three are different loci on the chromosome which consist the allele, capital C or small c, capital D or small d, capital E or small e. And it is also same. Student, these are the alleles, any one of the allele in these two, any one is present, and capital D, small d, any one is present, capital E or small e, any one is present. Okay, homologous chromosome pair 1. Okay, student, it is the one pair chromosome, first pair chromosome, showing three different loci with alleles per locus. Okay, student, each one of the loci that consists two alleles, in this only one is present. Student, genotypes, one capital C or small c, same students, same. Okay, one C or small c, one capital D or small d, one capital E or small e. This is shows by single chromosome. It is the single chromosome, it one is the another single chromosome. Next, genotypes. Which type of the genotypes that are enters into the next generation? Okay, which one of the genotype, which one is the allele that entered into the next generation that causes the presence of the genotype? Okay, like if you observe, these are the genotypes, like a capital, all are capital letters, C, D, E, all are small letters. Students, these type of the genotypes, this CDE is received by one chromosome, this is the CDE received by another one of the chromosome in children's. Next, students, if you observe, one capital C, one capital E, small d is received, and small c and capital D, small e, like this amount is also received. Anyone is in these two, anyone is received by the individual. In these two, one of the chromosome is received by the individual. This is the genotype is received by the individual. This is the genotype is received by the individual from the parents. This total amount of the genotype is received by the parents. Received by the parents. Student, if you observe in these genotypes, which is the genotype that causes the presence of the Rh factor in the individuals? Which type of the genotype that is received by the parents to the next generation that causes the presence or absence of the Rh factor? Student, in this genotype is with the dominant D allele, that means, student, if you observe dominant D allele, any one of the dominant D allele, student, it is the capital D, it is the capital D. Capital D is received by the children ones, received by the progeny from the parents, that causes the production of the Rh positive phenotype, that causes for the appearance of the Rh antigen, Rh factor. Any one of the dominant D, capital D, that is received by the individual from the parents, that is inherited from the parents to the next generation, that is the individual, that is the generation, that is the individual that shows the Rh antigen. Okay. Next, genotype is with double recessive genotype. double recessive DD. Student, from the parents, child receives double recessive D allele. Student, it is the small d, it is the small d. It is came from the one of the parent, mother parent, it is came from the father. Okay, because of presence of like these genotypes, small d is present. This is equal to recessive double allele, double D allele. Recessive double recessive D allele, DD. Student, one of the small d receives by the individual, receives by the child from the parent. Okay, that is the causes for the production of the Rh negative phenotype. Erythroblastosis fetalis. Already you know about Rh antigen. Rh antigen, that means Rh antigen, that is additionally another one of the antigen along with antigen A, antigen B. Rh antigen present on the plasma membrane of RBC. These are the individuals are called Rh positive. If you observe plasma membrane of RBC without the Rh antigen, these are the persons are called Rh negative. Okay. If you observe in the blood group of the different individuals, some of the individuals shows the Rh antigen, some of without the Rh antigen. Okay, so these are the blood groups by this called A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, O negative, like that. Student, erythroblastosis fetalis, also called hemolytic disease of newborn. Okay, and the student, if you observe O negative, these are the individuals called universal donors because of absence of all type of the antigens. In these individuals, Rh antigen is also absent. O negative. These individuals donate the blood to all type of the blood group persons so-called universal donors. AB positive, all type of the antigens present in these individuals, antigen A, antigen B, Rh antigen also. By this, these are the individuals receive all type of the blood groups from others, so-called universal recipient. It is also called hemolytic disease of newborn, hemolytic disease of fetus or newborn baby. Alloimmune condition, students, it is alloimmune condition. Alloimmune condition, that means immunity is produced in the body of individuals after the entry of non-self antigens. Non-self means that not related to the body parts. These are the antigens enters into the body from others, from outer surrounding. Okay, that is causes the production of the antibodies in the body. Entry of the antigens causes the production of the antibodies in the body. It is called alloimmune condition. It is also develops in Rh positive fetus. Developed fetus within the uterus, that the mother conceives pregnancy. In the uterus, within the fetus, that is the fetus is with Rh positive. Father Rh positive, student, if the father Rh positive, mother Rh negative. Okay, in this marriage that goes to the chance of the genetical incompatibility between the mother with Rh negative, developed fetus with Rh positive. Okay, it is very important, student. Growing fetus is with Rh positive and mother is Rh negative. Okay, fetal blood cells pass through placenta. Fetal blood cells, placenta is the barrier between the pregnancy mother and developed fetus. Fetal blood cells pass through the placenta into the, and these are contact with the, are entered into the blood circulation of the mother. Fetal blood cells, red blood cells entered into the mother circulation, maternal circulation through the placenta barrier at the time of the birth into Rh negative mother blood. Okay, student? At the time of the birth only these are the fetal blood cells enters into the maternal circulation. Next, mother is recognized Rh antigen gets sensitized. Students, at the time when these are the blood cells enters into the maternal part, mother part, that is the RBC is with Rh antigen. Okay, already about that, Rh positive fetus. Fetal blood cells with Rh antigen, that is the new antigen enters into the maternal circulation and immune system is identified and sensitized. Mother immune system is sensitized, identified and produces antibodies, produce Rh antibodies against the Rh antigen. These are based on the structure IgG type and these are smaller in all type of the antibodies. IgG type Rh antibodies entered into the fetal circulation through the placenta. By the placenta, these are the antibodies are enters into the fetus body, fetus circulation. First child is unaffected because fetus is delivered by time producing anti-D antibodies. Okay, before the production of the anti-D antibodies, before the production of the Rh antibodies, fetus complete its a gestation period, pregnancy period and delivers and goes to the paturation. Okay, within the, by the time of the production of the anti-D antibodies reaches into the fetal circulation, gestation period is completed and paturation is also completed. 9 months gestation period, pregnancy period is completed. Okay, and child delivers safely without the erythroblastosis fetalis, but maternal circulation, mother's body is with Rh antibodies. Student, if you observe, next during second pregnancy, second child is also with the Rh positive. Student, second child is with Rh positive, Rh antigen. Antibodies cross, antibodies are produced immediately and reaches the placenta and entered into the fetal blood, fetal circulation. Okay, student, and fetal blood cells are destroyed. Antibodies attacks the Rh antigen or red blood cells of the fetus. All the fetal blood cells are goes to the destruction. Okay, and causes erythroblastosis fetalis. Erythroblastosis fetalis. Okay, student. And what are the symptoms appeared in erythroblastosis fetalis? Symptoms are hemolytic anemia in fetus, developed fetus. Red blood cells are continuously goes to the destruction. Okay, that causes hemolytic anemia. Okay, by the destruction of the red blood cells, and bile juice is also goes to excess secretion and causes the jaundice. Bile juice secretes from the liver and causes the jaundice. Next, and the students, RBC for compensate for replacement of the decreased red blood cells, bone marrow and liver and the spleen. These are the three parts also produce excess amount of the red blood cells. These are also goes to the destruction and causes the anemia. Okay, and these are the red blood cells are produced in the form of the available in the form of the large immature erythroblast. Now about the prevention from erythroblastosis fetalis. Mother is Rh negative, fetus Rh positive. This is the Rh positive phenotypic character shows by the fetus, that is receives from the father. Okay, inherit. This is the gene which shows the character Rh positive, that is receives from the father. Okay, student. IgG anti-D antibodies are administered to the mother at the time of the 28th and 34th week of gestation. Okay, already you know about the gestation period, pregnancy period, that is 9 months. Okay, student, or that is equal to 38 or 40 weeks. Okay, student, at the time of the 28th or 34th weeks of gestation, mother's body is administered with the IgG type anti-D antibodies into the body. Okay, at the time of this genetical incompatibility between mother and the fetus. Okay, these are the antibodies introduced into the mother's body. These antibodies attacks the antigens and causes the destruction before the mother's immune system gets sensitized and produce the antibodies. Okay, Rh negative mother delivers, Rh positive baby delivers, Rh negative mother delivers Rh positive baby. Anti-D antibodies administered to mother soon after delivery. Soon after the delivery, these are the antibodies also administered into the mother's body. Okay, Rh negative mother delivers Rh positive baby. Anti-D antibodies administered to mothers after the delivery, soon after the delivery. Next, and develops passive immunity. Student, prevent formation of anti-D antibodies in the mother blood. Students, discuss about polygenic inheritance. Already you know about the erythroblastosis fetalis. Erythroblastosis fetalis, it is the one of the genetical incompatibility disorder between the Rh positive fetus and the Rh negative mother. Okay, at this type of the condition, Rh negative mother is administered with the for the protection of the erythroblastosis fetalis. Rh negative mother should be administered with the anti-D type of antibodies, Rh antibodies. Okay, just after the delivery. Okay students, after the delivery immediately should be administered with the Rhd type antibodies for the prevention of erythroblastosis fetalis. Okay, it is produces passive immunity. Passive immunity means these are the antibodies entered into the body through the injection, destroy all the antigens which are enters into the maternal circulation. Okay, by this way, by the production of the passive immunity, introducing of the ready-made antibodies which shows the effective action against the Rh antigens, okay, for the prevention of the erythroblastosis fetalis. Students, polygenic inheritance, it means additive effect of the two or more genes on single phenotypic character. Polygenic inheritance, it is opposite to the pleiotropy. Okay, additive effect of two or more genes, students, it is important, two or more genes on single phenotypic character. Single phenotypic character may be affected by, influenced by two or more genes. Students, what is quantitative characters? Quantitative character, that means phenotype depend on the action of many genes, also with the environment. One of the phenotype which is possessed by the individuals, that is by the action of the not a single gene, many genes, and also by the environment. Okay student, that is character, that is the phenotype present in the animals called quantitative characters. Quantitative variation, what is the meaning of quantitative variation? Existence of the different phenotypes individuals, different for only specific character, single character, that is shows in many forms. Existence of the different phenotypes, single character in the animals is appeared in the form of the different phenotypes. Student, that is called quantitative variations. For example, skin color and blood pressure. Student, blood pressure in the individuals, skin color in the individuals, this type of the variations, quantitative variations. Okay student, next, quantitative variation indicates polygenic inheritance. Next, skin color in man, it is the best example for polygenic inheritance. Polygenic, more genes. Okay, skin color in man, human skin color varies in population in gradients called quantitative character. Human skin color, it is the one of the character shows by the individuals, persons present in the population in gradients. Okay, that is called quantitative character. Students, if you observe skin pigmentation in humans, pigmentation of the skin is due to the deposition of the melanin, that is the pigment. Okay, skin pigmentation is caused by pigment in the skin. Okay, that is melanin. Okay, skin pigmentation in humans, this is the phenotypic character is by three genes students. Three genes, which are three genes? A, B, C are the genes. Okay student, skin pigmentation in humans, it is the phenotypic character. by the presence of the three genes. Okay, three genes that controls the single character, that is skin color. Okay, genes, which are the genes? Capital A, capital B, capital C are the genes. These are dominant alleles. These are the genes with dominant alleles in the form of these are the genes in the form of the dominant alleles. Capital A, capital B, capital C control the skin color in the human beings and responsible for dark skin color. Okay, student, dark skin color. Dominant alleles, these are the genes available in the form of the dominant alleles, and it is causes for the responsible for dark skin color. Next, small a, small b, small c, these are the alleles for gene. Okay, human skin color. And alleles for light skin color. Light, small a, small b, small c are the alleles for light skin color. Okay, student? And these are the alleles inherited from the parent to the progeny. Now, if you observe progeny, these are the genes are available. These are receiving from the parent individual, mother parent and the father parent. Okay, persons having all the capital letters, all the dominant alleles shows the phenotypic character of the skin color with very dark. Okay, and all the small letters, persons with phenotypic character very light skin color. Okay, capital A, small a, capital B, small b, capital C, small c, skin with intermediate color between very dark and the very light. Genotypes, capital A, capital B, small b, capital C, small c, and capital A, capital B, small c, small c. Okay, these are the progeny having three dominant alleles, capital A, capital B, small c, capital A, capital B. Three dominant alleles present in the individuals receiving from the parents, inherited from the parents to the next generations, having three dominant alleles. Okay, that makes the skin darkness in these individuals, skin darkness. Next, different skin color phenotypes, light skin, dark skin, intermediate color. Okay, due to the deposition, based on the deposition of the melanin pigment in the skin. Student, that could result from mating between capital A, small a, capital B, small b, capital C, small c. Okay, heterozygous organisms, individuals are which are shows these type of the genotype, individuals which shows these type of the genotypes having heterozygous organisms. Okay, why called heterozygous organisms? Having different alleles for gene, having different types of the alleles for the gene, that are individuals are called heterozygous organisms. Okay, number of each type of alleles in genotypes determine the darkness and the lightness of the skin in the individuals. Okay, it is about the polygenic inheritance with the example of skin color in human beings. Now discuss about sex chromosomes. What about the sex chromosomes? If you observe karyotype of the animal, total chromosome complement in cell of animal, there is a 6 chromosomes. What about the 6 chromosomes? 6 chromosomes are one pair chromosomes determination of the sex, helps in determination of the sex. In total chromosome complement, which are the chromosomes determine the sex? Called 6 chromosomes. These are one pair. Okay, sex chromosomes are called allosomes, students. Sex chromosomes are called allosomes. Student, two types of the chromosomes present in the karyotype. Okay, one pair sex chromosomes other than the sex chromosomes are called somatic chromosomes or autosomes. Autosomes, allosomes. Allosomes are sex chromosomes, one pair in number in animals. Still, inking. What about the role of the scientist? Inking in chromosomes, about the chromosomes. Student, inking identifies sex chromosomes from the experiment on the wasp. Inking goes to experiment on the wasp for identification of the sex chromosomes, identification of the sexual chromosomes. And observe spermatogenesis, students. Observe spermatogenesis. Student, spermatogenesis means production of the spermatozoa from the sexual organs, called spermatogenesis. In this process, spermatozoa or the sperm cells are produced from the organs, primary reproductive organs. Okay, and in this 50% of the spermatozoa receive one of the nuclear structure. Student, about in the formation of the sperm cells, 50% of the sperm cells is receive one of the structure, one of the nuclear structure, and under 50% did not receive it. Which are the spermatozoa, sperm cells that receive nuclear structure from the mother cell that is named with X body, that is inking, that is named with X body. 50% of the spermatozoa at the time of the spermatogenesis, these are the spermatozoa receive one of the nuclear structure from the mother cells present in the primary reproductive organs. Okay, that is the received structure named with the X body. X body is entered into the spermatozoa from the mother cell. Okay, and but he did not give about the importance of that X body, that nuclear structure. Okay, this is about the inking. Okay, student, and another. For example, students, at the time of this spermatogenesis, X0, okay, AA, it is the karyotype. It is the karyotype of the wasp. For example, student, this is the mother cell. This is the karyotype of the mother cell present in the wasp. Karyotype of the mother cell in the reproductive organ, primary reproductive organ in wasp. Okay, karyotype. It is the karyotype. Okay, it is the karyotype. Okay, mother cell. And at the time of the production of gametes, it is the mother cell goes to the meiotic division. Okay, and two type of the gametes are produces. Okay, these are gametes with AX and A0. Okay, student, these are gametes. These are gametes. Okay, student, in this, if you observe these two type of the cells, these two type of the sperm cells, okay, it is the mother cell. Mother cell goes to the meiotic division and produce two type of the cells. In these, students, capital A means It is the karyotype. Capital A means that is about that indicates the autosome complement. Autosomes complement. Okay, one of the capital A receives into the sperm cell, received by the sperm cell. Okay, that means in this total number of the autosomes which are present in the mother cell, in this half of the autosomes enters into the daughter cell. Okay, another half entered into the another cell. Okay student, in this is the X means X body. X means X body. Okay, after the going of the another scientist, the same type of the experiment going into the another scientist. After it is the X body is named with the X chromosome. Student, this is the X chromosome. X chromosome, it is the sex chromosome. Sex chromosome, it is the nuclear structure receives by the one of the chromosome, and another one it is does not receives it. That is the X body or X chromosome, that is the nuclear structure. It is not received by another gamete, another sperm cell. Okay student, this means half of the spermatozoa receives that nuclear structure or X body. Another 50% did not receive it. Hence Wilson identify Y chromosome as a sex determining chromosome in mealworm, Tenebrio molitor. Student, Stephen Wilson, after experiments on the mealworm, scientific name is Tenebrio molitor, firstly identify sex determining chromosome, that is the Y chromosome. Y chromosome is firstly identified by Stevens and Wilson. It is the sex determining chromosome. Next student, what about the homogametic? Homogametic students, for example, it is the karyotype of the mother cell. Okay, it is the karyotype of the mother cell. In this type of the karyotype, it is goes for the production of the gametes. Okay, it is the autosomal set in mother cell, autosome complement. Okay, autosome chromosome, these are sex chromosomes. XX means sex chromosomes, one pair sex chromosome present in the mother cell, XX. Okay student, in this AX and AX, okay, these are two gametes produces by the mother cell. Okay, all of the gametes is with AX and AX complement chromosome. Okay, these are similar, AX. Which one is the individual produce similar type of the gametes? Okay, that is the individual is called homogametic. Two sex chromosomes are similar, XX. Okay, individual gametes also similar. Okay, that is called homogametic. Heterogametic means, students, for example, if you observe AAXY, capital A means autosome complement present in the mother cell. Okay, XY, that means sex chromosomes are one pair. In this, one is the X, another one is the Y. Okay, XY type of the allosomes are present in this mother cell in the person. Student, it goes for the production of the gametes, half with AX complement chromosome, another half with AY complement chromosome. Okay student, in these two type of the gametes are produces. Okay, student, half with ax complement, another half with ay complement. Ax, ay. These type of the gametes produced by the person or the individual called heterogametic. Heterogametic. Two sex chromosomes different XY are contains only one sex chromosome X0. Okay, AA X0. It is also goes for example. Okay, students, and gametes are dissimilar. Student, that is called heterogametic condition. Heterogametic individual. Student, heterogametic sex determination. What is the meaning of heterogametic sex determination? Heterogametic sex means sex of species in which sex chromosomes are not similar. Student, if you observe male individual and the female individual, okay, if you observe the karyotype of both individuals, sex chromosomes are not similar, not present in similar. For example, AAXX male. Okay, student, AAXX male and AAX0 female. Okay, student, AAXX female, AAXY male, like that. Student, in this if you observe AAXX, okay, female one, AAXY male one. Okay, sex chromosomes are not similar in these two individuals. Okay, male and females does not possess same type of the sex chromosomes like XXX0, XXXY. Student, XXX0. This is the sex chromosomes. This type of the sex chromosomes present in the individual, that is called heterogametic sex determination. Okay, student, in these two type of the individual does not possess the similar type of the sex chromosomes. Chromosomal sex determination is by allosomes. Sex determination in the progeny, in the children's, that is depend on the sex chromosomes or allosomes, that is called chromosomal sex determination. Okay, student, in heterogametic sex determination, one of the sexes produce similar gametes and other sex produce dissimilar gametes, like this. Okay, in this one of the individual, one of the person produce similar type of the gametes, another one of the individual produce dissimilar type of the gametes. Okay, in heterogametic sex determination. Okay, student, and produce dissimilar gametes. Sex of the young one determined at the time of the fertilization. These are the gametes goes to the fertilization and produces the zygote. Okay, it is about the sex chromosomes and heterogametic sex determination. Students, today's topic is male heterogamety. Male heterogamety. Heterogametic male is present in this type of the sex determination. Sex determination, it is possible by the allosomes. Okay, students, male heterogamety means males are heterogametic. That means males produce two types of the gametes. Males produce dissimilar type of the gametes. Next, females homogametic. Females produces same type of the gametes, similar type of the gametes. Okay, male heterogamety, Okay, it is two kinds. There is two kinds. One is XXX0 type. Second one is XXXY type. Now what about the XXX0 type? Student, XX means these are the allosomes, sex chromosomes present in the females. X0, only one of the X chromosome as a sex chromosomes. Only one of the X chromosome is present in males. Okay, XXX0 type of male heterogamety or sex determination present in some insects, bugs, grasshoppers and cockroaches. Student, in this males, these are males, these are females. Okay, student, male one, female one. Okay, in this females karyotype is AAXX. This is the karyotype. Karyotype, that means this is the chromosome complement present in the somatic cell of the body. All the somatic cells in the body shows the karyotype with AAXX. Okay, AA, this is the AA means total the number of the autosomes in the cell, somatic cell. Next, XX means one pair. Already you know, if you observe the karyotype of the organism, one pair sex chromosomes are present. These are available as a capital X. Student, and if you observe male one, male, the karyotype is AAX0. Okay, X0, that means only one of the X chromosome is present in the karyotype of the somatic cell in males. Okay, student, gametes are produced from the female. Gametes are produced from the female. All are similar. Similar type of the gametes are produced by the AAXX karyotype female. Okay, all are with AX type. Similar type of the gametes. Next, another, male produces two types of the gametes. Okay, in this half with AX complement of the chromosome, another half with only autosomes without the sex chromosomes. Two types of the gametes are produced from the male. In this, half with AX complement, another half with only autosome. Okay, student, and in this type of the XXX0 type, in this type, sex determination of the children's or the offsprings, sex determination in the offsprings is determined by which type of the sperm cell, okay, that fertilizes the egg cell. Okay, student, it is about the XXX0 type of the sex determination or male heterogamety. Next, student, if you observe XXXY type, in these students, that is the sperm cell is only with the autosome, that is combines with the AX egg cell, and offspring is developed into the male one. Okay, if the sperm cell with the complement chromosome with the AX chromosomes, that is fertilize the egg cell and develop offspring is develops into the female one. Okay, student, XXXY type of the sex determination or male heterogamity is present in human beings, insects such as Drosophila. Student, in this females possess X karyotype, AAXX karyotype, and males possess AAXY karyotype. Okay. In this, males produce two types of the sperm cell, two types of the gametes. Half of the gamete is with X complement chromosome, another half with AY complement of chromosome. Okay, and females produces similar type of the gametes, similar type of the egg cell. These are all with X complement of chromosome. Okay, in this type of the sex determination, developed offsprings. Okay, sex determination in the offsprings is determined by which type of the sperm cell that fertilize the ova. Okay, student, this is about the XXXY type of male heterogamety. Okay, next student, and now goes to female heterogamety. What about the female heterogamety? In these, females are heterogametic. Females are heterogametic. That means females produces two types of the gametes. Students, these are the females. Females produce two types of the gametes. Okay, females are produce two types of the gametes in this also. So it is called female heterogamety. Males are homogametic. Males are homogametic. Males produces similar type of the gametes. Okay, students, aged only. Similar type of the gametes produces from the males. So these are available in males are homogametic. Males are homogametic. Males produces similar type of the gametes. Females produce two types of the gametes. Okay. So females are heterogametic condition. Next, in this is also available in two kinds. One is Z0 ZZ type, another one is ZW ZZ type. What about the Z0 ZZ type? Z0 ZZ type of the female heterogamety or sex determination is present in moths, example Fumia, and some butterflies. Okay, student, if you observe karyotype of the female AAZ0. Next, karyotype of the male AAZZ. Okay, students, because of presentation of the AAZ0 karyotype, females produce two types of the gametes. Half with aged complement of chromosome, another half with only autosome. Next, males produce similar type of the gametes, all of them with aged complement of chromosome. Okay, students, sex determination of the offsprings. Offsprings sex determination is by which type of the egg cell that fertilizes the sperm. Okay, student, in these two types of the ova or the egg cell, which type of the egg cell that is fertilizes the sperm? Okay, that depends the sex determination in the offsprings. Next, student, ZWZZ type. What about this type of the ZWZZ type of the sex determination or the female heterogamety is present in birds, reptiles, and some amount of the fishes. Student, in this, karyotype of the female is with AAZW. Karyotype of the males are with AAZZ. Okay, student, in this, females produce two types of the gametes, half of them with AZ, another half with AW chromosomes. Student, in this, males produce similar type of the gametes, all of the sperm cell is with AZ complement of chromosomes. Okay, student, and gametes are produced, goes to the fertilization and produces the two types of the offsprings, males and the females. Student, in this sex determination in the offsprings, the young ones, is determined with the which type of the egg cell that fertilizes the sperm. Okay, student, this is about the ZWZZ type. Okay, students, male heterogamety is with two types. XXX0 type present in some insects like bugs and grasshoppers, cockroaches. XXXY type present in human beings, insects such as Drosophila. Female heterogamety is of two kinds. Z0 ZZ type, example moths, fumia and some butterflies, and ZWZZ type present in birds, reptiles and some amount of the fishes. Students, sex determination in humans. Sex determination in humans, in this already you know about the heterogametic and homogametic, male heterogamety, female heterogamety. Okay, student, in this mechanism in case of humans is XXXY type. XXXY type. XXXY type is example for male heterogamety. Males having one pair of the sex chromosomes or the allosomes with the presence of the XY. One X chromosome, one Y chromosome. Okay, student. Next, if you observe number of the chromosomes in all the somatic cells in human beings, all the body cells in the human beings, there is a 46 chromosomes. Students, these are 23 pair of chromosomes. 23 pair chromosomes, that is the diploid cell, somatic cell, karyotype of the somatic cells, all the somatic cells in the body of human beings. There are 46 chromosomes, 23 pair chromosomes. Student, in this, the cells which shows the somatic cell which shows the 46 chromosomes, 23 chromosomes in the human beings, that is diploid cell. Okay, two sets of the chromosomes are present in the cell, that is called diploid. Okay, students, this diploid cell, that is the mother cell present in the human beings, that is goes to the meiosis, for the production of the gametes. Okay, Genesis. For example, oogenesis or spermatogenesis. These are the process for the production of the gametes such as sperm cell or the ova. Student, egg cell. This is the diploid cell goes to the meiotic division for the production of the gametes. Half of the chromosomes enters into the one cell, another half of the chromosomes are entered into the another gamete. Students, these two are gametes and chromosomes are 23 in number. Okay, half of the chromosomes present in the diploid cell enters into the each gamete. These two are gametes. These two are gametes. Gametes with a single set of the chromosomes. Student, mother cell that have the double set chromosomes. Hence it is called diploid cell. These are called haploid cells. Okay, student, these are the chromosomes, 23 in number, available in each gamete. Okay, these are ova, egg cell or the sperm cell. Okay, these are diploid cell, mother cell is the diploid cell, and daughter cells which are termed with the sperm cell or the egg cell is haploid cells. Haploid cell, diploid cell. Okay, student, In this 23 pair of the chromosomes, 22 pairs are same in both males and the females. Totally 23 pairs present in the diploid cell in males and the females. In these 22 pairs are same in both of the individuals, males and the females. Okay, student, these are the 22 pairs same in both males and the females are called autosomes. These are called autosomes. Okay, remaining one pair present in the male and the female, these are called sex chromosomes or the allosomes. If you observe female one pair of the X chromosomes, 22 pair of autosomes, and additionally with one pair, totally 23 pair. In these one pair, these are with XX chromosomes. XX chromosomes called one pair X chromosomes present in the females. And what about the males? Males have, with the presence of the one X chromosome, one Y chromosome. Okay, student, 23rd pair of the chromosomes available in the females as a XX chromosome, and in the males these are available in the form of the 1X and the 1Y chromosomes. Okay, student, if you observe spermatogenesis in the males, spermatogenesis, that means production of the sperms from the mother cells present in the reproductive organs. Okay, student, spermatogenesis, two types of the gametes are produces. Two types of the gametes. Student, it is the mother cell having the double set of the chromosomes. This is the diploid cell. Okay, two types of the gametes. Okay, one of the gamete is with the X complement chromosome, and the one is with AY complement of the chromosomes. 50% of the sperm carries X chromosome, another 50% of the sperm carries Y chromosome. Student, autosomes are similar in both of the sperm cells, both of the gametes, but if you observe sex chromosomes, okay, in this 50% of the sperm that carries the X chromosome, another 50% of the sperm that carries the Y chromosome. Okay, half of the sperms is with X complement chromosome, and the half with AY complement of chromosomes. And what about the females? Females produce one type of the ovum with X chromosomes. Student, these are females. This is the karyotype of the diploid cell in the females, AAXX. Okay, student, in this AAXX, if you observe the production of the gametes or the X cell, all the X cell receives X complement of the chromosomes. Student, all the gametes is with the X complement of the chromosomes. Hence, these are homogametic, these are heterogametic. In this case, ovum is fertilized by sperm. Ovum is fertilized by sperm carrying X chromosome. Zygote develops into female. Student, this is the ovum fertilized with sperm carrying the X chromosome. This is the sperm student. This is the sperm carrying the X chromosome. If the ova or egg cell is fertilized with the sperm which carrying the X chromosome, that is the offspring is develops into the female one. Next, if you observe another one of the case, fertilization of the ovum with Y chromosome carrying sperm, zygote develops into male offspring. Student, it is the ovum fertilized with the sperm which carrying the Y chromosome. This is the X chromosome carrying sperm. It is the Y chromosome carrying sperm. It is the ovum fertilized with the sperm that carrying the Y chromosome, the offspring is develops into the male one. Okay, student, develops into male offspring. Okay, student, Homo sapiens. Homo sapiens is the scientific name of the human beings. In pregnancy, always 50% have the 50% of the probability, either male child or female child. Okay, students, same probability that the offspring is develops into the male child or the female child. In this case, females are not blamed with the offspring develops into the female one. Okay, student. Y chromosome supports male sex. Y chromosome supports male sex because carrying of the that zygote with the Y chromosome. Okay, that is the Y chromosomes enters into the offspring, developed offspring or the zygote that is supports for the development of the secondary sexual character, and it is causes for appearing of the male sex. Y chromosome supports male sex as it has the genes for secondary sexual characters. Now discuss about genic balance theory of sex determination. Genic balance theory, students. In this, genes carried on sex chromosomes so that determine the sex of the offspring, sex of the individuals. Genes that are carried through the sex chromosomes. Okay, students, these are entered into the offsprings. Okay, that determines the sex of the individual. Okay, and XY are responsible for determining sex of offsprings. Okay, students, XY are the chromosomes, sex chromosomes in the male. Okay, and XX are the sex chromosomes or the allosomes in the female. XX, XY chromosomes. It is male heterogamety. Male produce two different types of the gametes. Okay, half with AX chromosome, another half with AY complement chromosomes. Student, but it is observed in male heterogamety, which are the example for XX, XY type of the sex determination, chromosomal sex determination. It is present in human beings and the Drosophila melanogaster fruit fly. But students, by this genic balance theory, sex chromosomes that determine the sex of the offsprings or the individual, it is not always true. Okay, student, and C.B. Bridges is the scientist, proposes a genic balance theory of sex determination. C.B. Bridges proposes a genic balance theory of sex determination after working on the Drosophila melanogaster fruit fly. Okay, he observed sex determination in the Drosophila melanogaster. Okay, student, and sex of the individual determined by balance between genes for the femaleness is located on the X chromosome, and genes for maleness locates on the autosomes. Okay, by C.B. Bridges, with the help of the genic balance theory, he proposes genic balance theory. By this, sex of the offsprings, sex of the individual in Drosophila melanogaster is determined by the two types of the chromosomes. In these, one is the X chromosome, another one is the autosome. Student, Y chromosome takes no part in the sex of the individual. Y chromosome, it is not determined the sex of the offsprings. Student, that is the offspring develops into the femaleness. The type of the genes are located on X chromosome. That is the offspring develops into the male one. The type of the genes locates on the autosomes, not a Y chromosome. Okay, and sex is determined by ratio of number of its X chromosomes and that of its autosome sets. Student, that is the ratio gives the sex of the offspring, determines the sex of the offspring. This is the ratio between number of its X chromosomes, which amount of the X chromosomes present in the zygotes that enters into the zygote, okay, from the gametes by the fertilization between two gametes, and number of autosomal sets that are entered into the zygote. Student, number of autosomal sets and number of X chromosomes, its X chromosomes. These are two important. Student, there is a sex of the individual determined by the ratio of the number of X chromosomes and number of autosomal sets. Okay, student, another sex index. He also gives one of the sex index that determines the sex of the offsprings. Sex index equal to number of X chromosomes by number of autosomes, X by A ratio. Sex index is equal to X by A ratio. That is the value gives the sex of the offsprings. Students, what about the non-disjunction? Non-disjunction, it is important, that determines the sex of the offsprings in Drosophila melanogaster. In this, what is the meaning of the non-disjunction? Non-disjunction, that means failure of the paired chromosomes to separate into the during the anaphase stage of the first and second meiotic divisions. Student, chromosomes are equally separated in the mother cell and distributed into the moves into the daughter cells, which are produced from the mother cell at the time of the meiotic divisions, first and the second meiotic division. Chromosomes are equally separated. Number of the chromosomes are equally separated into opposite poles. Okay, and moves into the daughter cells at the time of the meiotic divisions. Okay, in this non-disjunction, failure of the paired chromosomes to separate during the anaphase stage of the first and the second meiotic division. Okay, this causes for the production of the gametes with the abnormal number of chromosomes. Student, anaphase is the one of the stage in the cell division. Okay, that is the anaphase stage shows the movement of the equal number of the chromosomes into the daughter cells. Okay, failure of paired chromosomes to separate during the anaphase stage that leads to the production of the abnormal number of chromosomes in the daughter cells. Production of gametes is with abnormal number of the chromosomes. Okay, these are the gametes, abnormal gametes. Syngamy of such gametes with normal gametes, such type of the gametes, abnormal number of the chromosomes having gametes, such type of the gametes fertilized with the normal gametes. Syngamy of such gametes is with the normal gametes. produce zygotes with aneuploid karyotypes. What is the meaning of aneuploid? That is the cell having abnormal number of the chromosomes. Okay, does not shows the haploid set or does not have the diploid set. These are the cells shows abnormal count of the chromosomes. That is, the karyotype is with the abnormal number of the chromosomes is called aneuploid. 2n plus 1. 2n means diploid. Okay, double set of the chromosomes. 2n plus 1. Additionally, one chromosome is extra in that zygote, in that cell. 2n plus 1. 2n minus 1. 2n minus 1 means 2n means double set chromosomes. Minus 1. One chromosome is absent, less. One chromosome is less in that cell. That is aneuploid karyotype. Okay, student, if you observe, this is the female, this is the male Drosophila. Okay, due to the non-disjunction, failure of the separation of chromosomes into the daughter cell, this is the mother cell AAXX karyotype. Okay, student, AAXX female that goes for the production of the gametes. Okay, in this half of the gametes is with AXX complement chromosome, another half with only autosome. X chromosome is not present in this gamete, egg cell. Okay, student, two X chromosomes, one pair X chromosomes enters into the single gamete due to the non-disjunction. Okay, this is the abnormal gamete. Now it is called as abnormal gamete. This leads to the production of the aneuploid zygote. Okay, student, and this is the male one, male Drosophila, produce normal gametes, half with X chromosome, another half with AY chromosomes. These are the normal gametes fertilizes with the abnormal gamete. For example, AX complement chromosome, which are having AXX complement chromosome. Okay, that leads to the production of the gametes is with the aneuploid. Okay, students, in this AXX gamete or the egg cell fertilizes with the AY sperm cell, AY gamete. Student, that is the female receive, that is the zygote receives AAXXY chromosomes. Student, AX sperm cell that fused with the A0 egg cell, and that is the male offspring receives AAX0 complement chromosome. In this, AAXXY is develops, that is the offspring develops into the female, AAX0 offspring is develops into the male. In this, student, AAXXY fleece develops into fertile females. AAX0 flies develops into the sterile males. Okay, student, in this if you observe AAXXY offspring, this contains Y chromosome in its karyotype, but it is not develops into the male one. This is develops into fertile female. If you observe this karyotype, students, AAX0, okay, this is the karyotype is with the absence of Y chromosome, but is develops into sterile males. Okay, this is gives one of the information. Y chromosomes takes no part in the determination of sex of offspring. Y chromosome is present but not develop into the male. Y chromosome is absent but not develops into the female. Okay, This is concludes with the one of the information, that is Y chromosome takes no part in the determination of the sex. Student, in this fertile females means having ability to produce the offspring. That is, the young one have the ability to produce the offsprings, having ability to participate in the reproduction. Next, sterile males, these are the males loses its ability to reproduce. These are the males loses its ability to produce the offsprings, that is sterile males. Students, now going to discuss about the remaining part of genic balance theory of sex determination. Genic balance theory of sex determination in Drosophila, discovered by C.B. Bridges, Kelvin Black Bridges, American scientist. Okay, he goes to cross between triplid female having triple set complement of chromosomes, 3 autosomes, 3 X chromosome, triplid female, and with the diplid male, normal male, Drosophila, goes to cross between these two individuals, and he get different types of the individuals. Okay, observe offsprings, students, after this crossing between two individuals, triplid female, diplid male. Students, offsprings are available in diplid female, normal female, male, normal male, triplid female, intersexes. What is the meaning of the intersexes? Intersexes are the individuals or the Drosophila flies. Students, these are the individuals possess variations in sexual characteristics. Different types of the sexual characters are available in these individuals, called intersexes. Super males. Student, super males and super females. What about super males? Only one of the super males, that means only one of the X chromosome present in the individuals in the Drosophila. These are the super males, are the offsprings that receive only one of the X chromosome, having karyotype is with the only one of the X chromosome instead of XY chromosomes. These are individuals are termed with super males. Okay. Next, super males also shows the infertility. That means these are the individuals loses ability to produce offsprings. Next, super females. What about the super females? Super females means these are the women's. These are the female ones having greater strength than the normal females. Okay, and also loses its intellectual ability, loses its mental thinking, and some amount of the abnormalities is also shows by super females, having 3 X chromosomes instead of 1 pair X chromosomes. Okay, it is the one of the genetical disorder. Super males also one of the genetical disorder. Intersexes also one of the genetical disorder. Next, students, finally he concludes with Y chromosome in Drosophila. Y chromosome. What about the role of the Y chromosome? C.B. Bridges also explains that Y chromosome in Drosophila lacks male determining factor. Okay, male sex that is not determined by the Y chromosome. Okay, why that is not determines the male sex? Because Y chromosome lacks male determining factor. male determining factor, that is also called testis determining factor, TDF. Okay, presence of this TDF, male determining factor, on the Y chromosome, this causes for the development of into the, that is the offspring is develops into the male one, but Y chromosome does not shows or lacks the male determining factor, that TDF. Okay student, it is a TDF, male determining factor. It is encoded by one of the gene, that is called SRY gene. SRY gene encodes the testis determining factor, male determining factor, that causes the offspring develops into the male one or male sex. SRY gene, it is also called sex determining region Y. Okay, this entire the gene is absent on the Y chromosome. Okay, and it is also lacks the male determining factor, testis determining factor, because if Y chromosome takes no part in the determination of the male sex. Okay, but Y chromosome in Drosophila are required for male fertility. Okay student, next it is the table that shows the different sex chromosome complement that receives by the offsprings or the zygote, and applied sets of the autosomes present in the zygote or the offsprings, and X by A ratio, this is the X by A ratio value, and according to the X by A ratio, that is the which type of the phenotype present in the offsprings. These are sexual phenotypes. Student, for example, sex chromosome complement that is received by the zygote appeared in the zygote after the fertilization between the ova and egg cell. Student, karyotype is with XXAA. Two autosomes present in the zygote, okay, and two X chromosomes. What about the X by A ratio? 2 by 2. X by A means number of the X chromosomes, 2 in number. Number of autosomal sets, students, 2 by 2. That value, X by A ratio value is 1.0. That is the offspring develops into the female. Okay, CB Bridges also gives sex index, that means X by A ratio, that determines the sex of the individual. Okay, X by A ratio is equal to 1, that is the offspring sexual phenotype is female. Next students, if you observe XY karyotype and 2 autosomes, sex chromosomes are XY and 2 autosomes. Okay, that is the offspring is develops into the male one, and ratio X by A ratio is equal to 0.5. That means X chromosomes are 1 in number, and autosomes are 2 in number, 1 by 2, that is equal to 0.5. 0.5 value, that is the offspring develops into the male one. Okay, X0AA, that is equal to X by A ratio is 1 by 2, that is X by A ratio is 0.5. Offsprings developed into male. Okay, XXY, these type of the chromosomes received by the zygote. Okay, and 2 autosomes, 2 by 2. X by A ratio is equal to 1, that is the offsprings develops into the female. Next, 3 X chromosomes, 2 autosomes that are present in the zygote after the fertilization between the egg and the sperm cell. Okay, students, these are abnormal gametes. These 3 are abnormal gametes. X0 also, these are the abnormal gametes, students. Okay, 3 X chromosomes and 2 autosomes present in the zygote that is the X by A ratio is 3 by 2. 3 by 2 equal to 1.5. 1.5 that is develops into the meta female. Okay, 3 X chromosomes and 2 autosomes. X by A ratio is equal to 1.5. Offsprings develops into meta female. Next, 2 X chromosomes and 3 autosomes, 2 by 3. X by A ratio is equal to 0.7. That is the offsprings develops into the intersex. Next, only one of the X chromosome and 3 autosomes. That is 1 by 3. Okay student, 1 by 3 that is equal to 0.33. Offsprings develops into meta male. Meta male also called super male. Meta female also called super female. Next, 4 X chromosomes and 3 autosomes received by the zygotes. These type of the zygote is produced due to the occurrence of the non-disjunction at the production of the gametes. Okay students, and in this type of the X by A ratio, the value is 1.33. That is the offspring is develops into the meta female. Is X by A ratio is equal to the 1, that are the offsprings are develops into female. Okay, if the ratio is equal to X by A ratio is equal to 0.5, that are the offsprings are develops into the male. Okay students, and if the ratio is greater than 1, the ratio is greater than 1.5 greater than 1, that are the offsprings are develops into the meta female also called super females. Okay, super females are infertile. Okay, next, if the ratio is less than 0.5, the ratio is X by A ratio is less than 0.5, that are the offsprings are develops into the meta male also called super male. Okay student, ratio between 0.5 and 1.0, 0.5 and 1.0 students, 0.5, 0.67 is the ratio between 0.5 and 1.0, 0.67, that are the offsprings are develops into intersex. Okay student, it is about genic balance theory of sex determination by in the Drosophila by C.B. Britt. Students, now going to discuss about bar bodies. Okay, what about the bar bodies? Okay, condensed inactive X chromosome found in nuclei of all somatic cells in the body of most female mammals, including humans. That is goes to the chromatinization, that is the sex chromatin. Students, females have 2 X chromosomes. Males have XY chromosomes. So for example, if you observe human beings, okay, females have 2 X chromosomes, males have only one of the X chromosome. Okay students, in this females additionally, when it is compared with the sex chromosomes, X chromosome present in the male, females have 2 X chromosomes. Students, for the dosage compensation, one of the X chromosome present in the females goes to the condensation and goes to the inactive. Okay, that is the condensed inactive X chromosome that is found in the nuclei of the somatic cells in most mammals, including human beings also. that is called bar bodies. That is called bar bodies. Extra X chromosome present in the females, that is goes to the inactivation and the condensation, and which is found in nucleus of the somatic cells in most mammals and the human beings. Student, nuclei, it is the plural form of the nucleus. Nucleus is the singular, nuclei is the plural. Okay. And what is the meaning of heterochromatinization? Heterochromatinization. Student, nucleus that consists of euchromatin enter the part of the DNA, RNA and protein content that goes to the expression, that goes to the gene expression, that codes the formation of some amount of the materials, that is called euchromatin. That is called euchromatin. Okay, it is active form of the genetic material in the genome of the nucleus, which is the content that goes to the gene expression. Active form of the genome, that is called euchromatin, present in the nucleus of the cell. Next, conversion of the euchromatin into heterochromatin. Heterochromatin, inactive form of the genome, that is opposite to the euchromatin. Okay, student, heterochromatin, that is the content of the DNA does not goes to the gene expression, that is called heterochromatin. Okay, conversion of euchromatin into heterochromatin during inactivation of the X chromosome in early stage of the embryogenesis, early stages of the embryonic development. Euchromatin, some amount of the euchromatin present in the genome within the nucleus of the cell, that goes to the, that convert into the heterochromatin. Okay, student, at the time of the inactivation, during the inactivation of the X chromosome, it is conversion of the euchromatin into the heterochromatin. The entire the DNA is attaches with the one X chromosome, which is participates in the gene expression, that is goes to the inactivation of the X chromosome. Okay, student, in early embryonic stages of the mammalian females. Okay, students, X chromosome present in the female, one of the X chromosome present in the female, which is the DNA is attaches with the X chromosome, that entire the euchromatin goes to the heterochromatin during the inactivation of the X chromosome in early embryonic stages of the female mammals. Student, that is called heterochromatinization. Okay, student, heterochromatinized X chromosome, this is the X chromosome before it going into the heterochromatinization, that is available in the form of the euchromatin in entire genome of the nucleus of cell. That is, heterochromatin is heterochromatinization. After the heterochromatinization, heterochromatinized chromosome, heterochromatinized X chromosome is appeared as a darkly stained body. Student, that is heterochromatinized X chromosome appeared as a darkly stained body that attaches to the nucleus membrane. Okay, student, that is discovered by, identified by Murray L. Barr. Hence these are called bar bodies. one of the X chromosome, it is goes to the condense, it is goes to the inactivation. Okay, it goes to the heterochromatinization. Okay students, heterochromatin, it is converted into the heterochromatin. This is appeared as a dark body which is attaches to the nucleus membrane of the cell. Sure, that is called heterochromatinization. This entire the process is firstly identified and discovered by Murray L. Barr. Hence it is called Barr bodies. Student, Barr and Bertram, two scientists, Barr and Bertram. These are the Barr bodies observed in nerve cells of the female cats, and these are at the same time, these are not present in the males. These are the Barr bodies observed in the somatic cells of the female cat, and at the same time male cat does not possess these Barr bodies. Okay, in humans identified female cells, these are the Barr bodies in humans identified in the female cells and also derived from the buccal cells present in buccal mucosa, and also derived from the fibroblast. Fibroblast are the cells present in the connective tissue of the body. Okay, in female mammals, these are the Barr bodies are appeared as a drumstick like structure and attaches to the any lobe of the nucleus of neutrophils. It is appeared as a drumstick like projection on one of the lobe of the neutrophils. Neutrophils are the one type of the white blood cells, and nucleus is divides into lobes. Any one of the lobe passes the drumstick like projection, that is the Barr body. Okay student, and another one, Mary Lyon and Lyon Russell. Okay, two scientists proposes X inactivation, also called lionization. Okay student, what is the meaning of the X inactivation or lionization? That means two copies of X chromosomes present in the females. In this, one of the X chromosome present in body cells of the female mammals goes to the inactivation. Two X chromosomes present in the females for the dosage compensation. Okay, for the equalization of the gene expression in both of the individuals, male and the females. One of the X chromosome present in the females, that is appeared, that is goes to the conversion of the heterochromatin, goes to the condensation, goes to the inactivation, and appeared as a dark body like structure on the nucleus membrane of the cell. That is called X inactivation or lionization by Mary Lyon and Lyon Russell. Student, what is the meaning of the dosage compensation? Dosage compensation, that means, students, organisms equalize expression of the genes between two different individuals, so two different biosexes, males and the females. Okay, to equalize the expression of the genes in two different males and the females. Okay, student, females have two X chromosomes, males have only one X chromosome. Okay, by the processing of the gene expression, one of the X chromosome shows the gene expression, and another one of the X chromosome also shows the gene expression available as a euchromatin. Okay, but males have only one of the X chromosome. okay, which is available in the form of the euchromatin. In this, to equalize the gene expression in both of the individuals, females which are having two X chromosomes, one of the X chromosome is goes to the inactivation, okay, goes to the condensation, goes to the heterochromatinization. Student, that is called dosage compensation. Student, organism equalize expression of the gene expression between two different individuals. In humans, females which have two X chromosomes silence the transcription of one X chromosome. One of the X chromosome, by going into condensation, inactivation, heterochromatinization, one of the X chromosome is enters into the silence to gene expression, to transcription. Okay, students, females silence the transcription of one X chromosome, okay, of each pair, and all information is transcribed from another one of the X chromosome, that is equal to the males. Okay, by this way, thus gene expression in the females is equal to the males. Okay, students, this random inactivation of one X chromosome, student, females have two X chromosomes. This is the one of the X chromosome, this is the one of the X chromosome. Okay, after the heterochromatinization, one of the X chromosome in the females going to inactivation, going to condensation, going to heterochromatinization. Student, this is X chromosome, small X chromosome. After conversion of the heterochromatinization, after the condensation, after the inactivation, student, that is equal to the gene expression of males. Males have only one of the X chromosome. After the chromatinization, after the heterochromatinization, females also possess one X chromosome. This is available in the form of the bar body. Okay, this is the Y chromosome. Okay, gene expression is equal after the condensation, inactivation in both of the individuals, such as males and the females. Females have one X chromosome, another one of the X chromosome is available in the form of the bar body, and males have only one of the X chromosome, another one is the Y chromosome. Students, if you observe, this is the body cell, somatic cell membrane, and it is the nucleus. Student, nucleus, it is the entire cytoplasm. Nucleus membrane, if you observe nucleus membrane, the bar body is attaches. Bar body is appeared as a dark body like structure and attaches to the nucleus membrane. Okay, student, this is the bar body. This is the heterochromatinization form of the X chromosome. X chromosome is appeared as a bar body after the heterochromatinization. Student, if you observe, number of bar bodies follows as n-1 rule. Okay, how to discover the number of bar bodies in the somatic cells? Number of bar bodies identified as a following of n-1 rule. n-1 rule that helps to identification of the number of bar bodies in the somatic cell. Student, if you observe Turner's females means this is the Turner syndrome present in the females. It is the one of the genetic disorder, Turner syndrome. Okay, 45 chromosomes, totally 45 chromosomes present in the karyotype. karyotype is with one absence of one X chromosomes. Females possess two X chromosomes. If you observe in Turner's females, one of the X chromosome is absent. Okay, students, and total the number of the chromosome is equal to 45. Okay, instead of 46 chromosomes, one of the X chromosome is absent. Okay, student, and n-1 that is equal to n-1. n-1 means n is equal to total number of X chromosomes. Total number of X chromosome is 1 minus 1, that is equal to 0. So no bar bodies present in Turner's females. Okay, how many number of bar bodies present in Klinefelter males? Klinefelter syndrome, it is the one of the genetical disorder. Okay, karyotype is 47XXY. Normally 2 X chromosomes present in the females and XY chromosomes present in the females. Student, Klinefelter males possess one X chromosome additionally. Student, total number of the chromosomes are X chromosomes are 2 in number. Students, according to n-1 rule, 2 minus 1 equal to 1, number of bar bodies are 1. And females possess 47 chromosomes totally with karyotype XXY. 3 X chromosomes present in the females. If 3 X chromosomes present in the females, number of bar bodies are 3 minus 1 equal to 2. Number of bar bodies are 2 in number. And 48 chromosomes, total 48 chromosomes are present and X chromosomes are 4 in number. Okay, and n-1 rule equal to 3. Students, 4 minus 1. Number of X chromosomes are 4. 4 minus 1. Total 3. Number of bar bodies are 3 in number. Okay, number of bar bodies identified as n-1 rule. n-1 rule. N means n is equal to total the number of the X chromosomes minus 1. Students, 2 minus 1, number of bar bodies is 1. 3 minus 1, number of bar bodies are 2 in number. And 4 minus 1, number of bar bodies are 3. Okay, students, in this way, number of bar bodies are identified in the somatic cells of the most mammals. Students, now discuss about Haplodiploidy. Haplodiploidy, it is the one type of the sex determination present in honeybees. Students, mechanism of sex determination present in Hymenopteran insects. Hymenoptera, it is the one of the order present in the phylum Arthropoda. Okay, Hymenoptera includes with insects. In these animals, sex determination, mechanism of the sex determination is called Haplodiploidy. Students, which are the animals present in Hymenoptera? These are the insects like honeybees, ants and wasps. Okay, in these animals shows the Haplodiploidy. Sex of offspring determined by number of sets of chromosomes that receives. Offspring develops into the male one, offspring develops into the female one. That is depend on the receiving of the number of chromosomal sets from the parents. Okay, student. In this, fertilized egg develops into the females. Fertilized egg, student, that is the egg fertilized with the sperm. That is called zygote. Fertilized egg develops into offspring, that are the offsprings, develops into the females. Okay, females have double the number of the chromosomes, that is diploid. All the somatic cells in the females, body of females, having double number of the chromosomes, that is called diploid. Okay, fertilized egg develop into the females, that is important, having double the number of the chromosome in each somatic cell of the body. Unfertilized egg, that is the egg is not fertilized with the sperm. Okay, unfertilized egg is also develops into the young one. Okay, unfertilized egg develops into the young one, that is called males or drones. Okay, males drones produces from developed from unfertilized egg, that is the process is called parthenogenesis. What is the meaning of the parthenogenesis? Unfertilized develops into the offsprings. Unfertilized egg growth into the young ones, that is the process of the reproduction is called parthenogenesis, which is available in hymenopteran insects. Student, these are the half number of the chromosomes. Students, males that have applied, not a diploid, applied. Students, these are not fertilized. These are not fertilized. So all the somatic cells present in the males or the drones shows the half number of the chromosomes. Of the number of the chromosomes, that is called applied. Applied available in body cells, somatic cells of the males. Diploid is available in somatic cells of the females. Fertilized egg develops into the females. Unfertilized egg develops into the males, also called drones. Next, honeybee, that is the scientific name is Apis mellifera. Apis mellifera, it is the scientific name. Okay, in these drones, queens and worker bees are separately available in the colony. Drones having applied number of the chromosomes. Entire the chromosomes present in the cell, that is equal to 16 chromosomes. 16 chromosomes present in each somatic cell of the drone. Okay, next queens and worker bees that possess 32 chromosomes in nucleus of each somatic cell. Diploid, that is called diploid. Double the sets of the chromosomes present in somatic cells, diploid, in queens and worker bees. Of the number of the chromosomes present in somatic cells of the drones, that is called applied. Drones shows applied, queens and worker bees shows diploid. This, students, males produce sperms. Males produces sperms by mitosis division. Students, in these drones, also called male ones, these are the drones who produce sperm cells by the process of the mitosis. Entire the genome present in the drones or the males that are entered into the sperm cells. Okay, number of the chromosomes are 16 in number. After the formation of the sperm cells, these are also shows the 16 chromosomes in each cell, because these are formed by mitotic division, and entire 100% of the genes also present in the sperm cells. Because of these are the sperms formed by produced by mitotic division. Entire the genome present in the somatic cells of the drones are entered into the sperm cell. Total the genes, 100% of the genes enters into the sperm cell. Okay, males produce sperms by mitosis. So males have no fathers. It is important, students. Males have no father. and that cannot have sons. Males cannot have sons. Okay, and but have grandfathers and the grandsons. Okay, student, because of the haplodiploidy. Next, students, and the female that feed royal jelly. What is the meaning of the royal jelly? Royal jelly, it is the nutrition for the larvae. It is the food for developed ones in the colony. And this is the royal jelly is secretes from the hypopharynx of the worker bees. Okay, it is a nutrient rich food that is feeded by the one of the larvae that is develops into the next queen in the colony. Female that feed royal jelly becomes the fertile queen. Which one is the larva that is feeded with the royal jelly, which is secretes from the hypopharynx of the females, that is develops into the fertile queen. Okay, student, what about the queen? Queen having diploid chromosome, 32 chromosomes in each somatic cell. Other female that feed honey, pollen and becomes into sterile females. These are called worker bees. In each of the colony, 3 types of honeybees are there. These are worker bees, and the worker bees are sterile females. And fertile queen, only one of the queen. And other students, drones. Drones are also called males. If you observe, students, queen bee that is mated with the drones. For example, offsprings, female offsprings, female progeny shares 150 genes out of the total 200 genes, 200 possible genes. Okay, students, female progeny receives or shares 150 genes in total out of the 200 possible units, 200 genes. Students, female queen bee, female also called queen bee. Okay, this is the queen bee produces eggs. These are the eggs are applied. Okay, females queen bee that is goes to the meiotic division and produces eggs. Eggs are applied and having half the number of the genes. 50% of the genes receives by each of the egg, so that is called applied. Students, if you observe drones produce sperms by going on mitotic division. Enter the genome present in the somatic cells of the drones, that is entered into the sperm cell. So 100% of the genes are carried by the sperm cell. Okay, this type of the eggs fertilized with the sperms. Fertilized egg, students, fertilized egg develops into the females. Okay, female progeny that shares 150 genes, 50% and 100%, 150 genes out of possibility of 200 possible genes. Okay, student, it is haplodiploid. Students, now today's topic is sex linkage in Drosophila. Student, what is the meaning of sex linked inheritance? Inheritance of the trait from the parents to the progeny by the one of the gene present on the locus of sex chromosomes. Okay, that is the gene inherited from the parents to the next generation. That is also leads to the inheritance of the traits. That is the characteristic feature, that is the trait, that is the phenotypic character which is available in the next generation. That is caused by the inheritance of the responsible gene through the sex chromosome. that is called sex linked inheritance. It is totally different from the inheritance of the genes from the autosomes. Okay student, next, sex linkage in Drosophila. This means inheritance of the trait that determined by the gene locates on the one of the sex chromosomes. Already students, you know, one pair sex chromosomes, one pair allosomes present in the Drosophila or human beings. These are the sex chromosomes is with the genes. These are the genes present on the or locates on the locus of one of the sex chromosome. Any one of the sex chromosome that shows the gene that is the causes for the appearance of the phenotypic character that is inherited. That is the inheritance from the parents to the next generation is called sex linked inheritance. Okay, which one of the progeny or the offsprings that receives the chromosome which shows the genes that is caused for the appearance of the phenotypic trait, that is the responsible offspring shows the same type of the phenotypic character. Okay, sex linkage in Drosophila, it is introduced by T.H. Morgan in 1910. Thomas Hunt Morgan. Okay student, okay, what about his experiment, his analyzation on the Drosophila melanogaster? Students, in Drosophila, sudden appearance of the one white-eyed in culture of a wild red-eyed Drosophila. If you observe students, Drosophila eyes color is red in color. Red colored eyes, it is the phenotypic character by the Drosophila. It is the wild type, normal character shows by the Drosophila. In the culture of the Drosophila, wild type red eye colored Drosophila, suddenly he observed some amount of the Drosophila shows the white colored eyes. Okay, that is the one of the character he identified. T.H. Morgan identified that is the phenotypic character. Students, and another, Morgan crossed with a white-eyed male. Morgan crossed with a white-eyed male with a red-eyed female. Student, white-eyed male, this is white-eyed male, this is a red-eyed female. Two are Drosophila. Okay students, T.H. Morgan crosses between two individuals, two Drosophila. Male is a white-eyed male, female Drosophila is red-eyed. Okay student, red-eyed, that means X capital R. Okay, both of the allele are available in dominant position. Okay, X chromosome is with gene or dominant allele. Okay, another one of the X chromosome in female Drosophila also has dominant allele, that is capital R. Capital R causes for the appearance of the red colored eyes in Drosophila. Next, if you observe white-eyed male, students, X, okay, with the allele, that is the allele is recessive. X small r, that is the responsible gene which causes the color of the eyes, that is present on the locus of X chromosome. Student, it is not available on the locus of Y chromosome. Okay, which causes the white eye color, which causes the red eye color, the responsible gene present on the locus of X chromosome. available in the form of the recessive allele or the dominant allele. Third is X smaller y. Y is without alleles because of it is not shows the any type of the genes that causes for appearance of the white eye or red eye, that is the phenotypic character. Because of Y is with the absence of that genes. Okay student, and after Morgan observed F1 generation. All the F1 generation is with red-eyed females, and another one red-eyed males. Students, F1 flies were red-eyed, all are red-eyed. F1 flies were self-crossed red and white eye individuals appeared in 3:1 ratio, no female with white eye. Students, which are the individuals are available in the, which are the offsprings available in the F1 generation? And Morgan crosses between these two individuals. These are red-eyed female and students, these are red-eyed males. And at the same time he also get F2 generation. If you observe F2 generation, these are the F2 generation individuals, F2 generation offsprings. Students, these are the gametes X capital R, A capital R. All gametes are similar, X capital R. Okay, one is the X chromosome is X recessive allele smaller, another one is Y chromosome. Because of two types of the gametes are produced, of them with X smaller, another of with only Y. Okay students, and F1 generation is available between the F1 generation, and Morgan goes to the crossing, and 4 offsprings are available in the F2 generation. In the F2 generation, students, white eye individuals and red eye individuals are available in 3:1 ratio. Okay students, red-eyed female, red-eyed male. Okay, these two are females, these two are males. Okay, females are with red eye and males, one of the male, 50% of the males is with red eye, another 50% of the male is with white eye. Okay students, no female with white eye. Females are, if you observe offsprings, if you observe F2 generation individuals, no one of the female is with white eye color. Okay, and Morgan finally concluded with gene for eye color, which is the gene for the eye color present on the loci of X chromosome, locates on the X chromosome. Okay student, it is the final conclusion of the T.H. Morgan. T.H. Morgan finally concluded with that is the gene responsible for color of the Drosophila is present on the locus of X chromosome. Okay student, this is firstly observed by T.H. Morgan in 1990 in the culture of the Drosophila melanogaster. Next students, fleas will have white colored only X smaller. Okay student, there is no female is with homozygous recessive. Student, homozygous recessive, that means both of the X chromosomes in the female having the recessive allele. X chromosomes. X chromosome is with recessive allele. Another one of the X chromosome is also with the recessive allele. Both of the X chromosomes have the recessive allele that causes for the white eye color in the females. Students, fleas will have white color only X smaller, homozygous recessive. Okay, students. Next, another male carries a gene for white eye. Male carries a gene for white eye, then individual will be white-eyed male. Student, if you observe white-eyed male, that means X, Y are the chromosomes. XY are the chromosomes. Student, in this, males carry gene for white eye. Then individual will be white-eyed male. Which one is the responsible for white colored eyes? That is the X with recessive allele. X that consists recessive allele. X smaller. Only one of the X chromosome present in the males that is with the recessive allele. That causes for the appearance of the white colored eyes in the males. But females, when it is shows the white colored eye, both of the X chromosomes having the recessive allele. That causes for the phenotypic character, that is white colored eyes in the females. Okay. Next, inheritance of this X chromosome occurs in a crisscross manner. Student, it is the white-eyed responsible gene present on the loci of X chromosome in the males that is entered into the F1 generation and it is appeared through the red-eyed female. Okay, student. And it is the XR gene. This is the responsible gene that causes for the red-eyed. And this is the gene, student. This is the gene which is received from the father. Okay. This is entered into the F1 generation and available in the form of the white-eyed male. Student, it is the gene. Okay, students. This is called crisscross pattern of the inheritance. And he also goes to reciprocal cross between these two type of the individuals, white-eyed female and red-eyed male. So opposite sexes, which shows mainly the opposite phenotypic character. White-eyed female, red-eyed male. Okay, students, in this X smaller that causes the white eye. Okay, and X capital that causes the red eye. Both of the X chromosome is with the recessive allele that causes white colored eye. Okay, F1 generation. This is the F1 generation. Two types of the different individuals are available in F1 generation: red-eyed female and white-eyed male. Okay, this is the white-eyed character appeared in the males because he receives X recessive allele. X is with the recessive allele that is received by the male ones. Okay, and this is the X smaller, X with the recessive allele that is transmit to the female one, which is available in F2 generation. Student, it is the X recessive allele which causes the white eye. This is the female one transmit to his son. Through his son, that is transmit to the his granddaughter. His granddaughter available in the form of the white-eyed female. Students, both are different. Okay, it is the crisscross pattern of the inheritance. It means male transmits his character to his grandson. In this first one, male transmit his character, white colored eye phenotypic character, to his grandson through his daughter. Next, in the reciprocal cross, students, while female transmits, female one, that is the X smaller receive allele, that is the phenotypic character, transmit to his granddaughter, by his son. Okay, students, it is the sex linkage in Drosophila. Sex linked genes. That means genes locates on the loci of X chromosome and the Y chromosomes. Okay, that is called sex linked genes. Genes present on X chromosome or Y chromosome. Phenotypic character that is shown by the offsprings or the young ones, causing responsible genes or the alleles present on the locus of X chromosome and the Y chromosome, that is called sex linked genes. It is with X linked gene, Y linked gene and XY linked genes. In this X linked gene means genes locates on X chromosome whose that same type of the alleles are absent on Y chromosome. Okay, student, if you observe one of the gene, the type of the alleles present on the loci of X chromosome only, same type of the genes are absent on Y chromosome, that is called X linked gene. Gene linked with the X chromosome, gene present on the loci of X chromosome. Okay, and in this females are homozygous and heterozygous. Okay, what is the difference between about the X linked gene? Females are homozygous, that means one of the gene that is available in the form of the two alleles, one is the recessive and the one is the dominant. Both of the X chromosomes in the females having the same type of the allele, same type of the dominant allele are also available in this form. Both type of the X chromosomes having the recessive allele on the loci of X chromosome, that is called homozygous. Next, heterozygous, that means both of the X chromosomes having the different allele. That means one of the X chromosome is with the dominant allele, another one is the recessive allele. That is called heterozygous. It is the condition that is called heterozygous for that type of the gene. This is the condition available, that is called homozygous. Next, males are hemizygous. What is the meaning of hemizygous? Because of hemizygous caused by the only one of the X chromosome and one Y chromosome in the males. That is the gene available on the loci of X chromosome only, because it is X linked gene. Y chromosome that not shows that type of the gene. X with the dominant allele, Y chromosome. X with the recessive allele and also with the Y chromosome. This is the condition or this condition available in the males, that is called hemizygous. Okay, hemizygous is the condition occurs due to the presentation of the only one X chromosome, another one is the Y. Next, Y linked genes. Y linked genes means the type of the genes locates on the Y chromosome and same type of the alleles are absent on X chromosome. Okay, the type of the genes linked with the Y chromosome, that is also called holoandric genes. Students, X chromosome is without the gene, Y chromosome is with the gene available in form of the recessive allele or dominant allele. Okay, genes linked with the Y chromosome, that is called Y-linked genes or coandric genes. Next, males are hemizygous. Males are hemizygous for these alleles because of only one of the Y chromosome present in the males. Already you know about the hemizygous. Males are hemizygous for both type of Y-linked genes and the X-linked genes. Next, these do not undergo pairing during meiosis. Students, Y-linked genes do not undergo pairing at the time of the meiotic division, okay, because of presentation of the only one of the Y chromosome. Next, XY linked genes. XY linked genes means that are the genes are linked up with the X chromosome and the Y chromosome. Genes locates on pseudo-autosomal regions of the X and Y chromosome. If you observe entire the part of the X chromosome and the Y chromosome, some amount of the genes, some amount of the regions on X and the Y chromosome are consist for regions, pseudo-autosomal regions. These are the regions of X and the Y chromosomes are behave like a autosomes. Okay, these type of the genes are also called incompletely sex-linked genes. Okay, these are the genes also undergoes to the pairing during the meiotic division of the cell. Student, X chromosome is with the gene, Y chromosome also with the genes. Okay, and X chromosome is with the dominant gene, Y chromosome is with the recessive gene. These type of the genes available and linked up with the X and the Y chromosome is called XY linked genes. Students, now discuss about X-linked recessive inheritance. X-linked recessive inheritance means one of the character, one of the phenotypic trait inherited from the parents to the next generation because that causing gene, responsible gene locates on the X chromosome in the form of the recessive allele. Okay, male hemizygous, only one X chromosome is present. Okay, male hemizygous means, student, it is the X chromosome, it is the Y chromosome. Okay, X chromosome is with the recessive allele, that is enough to appearance of the phenotypic trait. Okay, next, females homozygous. Females homozygous, that means both of the X chromosomes, it is the X in females. Both X chromosomes have the recessive allele. Okay, recessive allele. Student, that is causes the appearance of the phenotypic trait, phenotypic character in the females. Okay, males hemizygous, females homozygous, that this is the condition is enough to express the type of the phenotypic character. Okay, affected males get this allele from mothers. Students, mothers, if you observe mothers, affected males get this allele from mothers. Students, if you observe, he is the affected male having the X chromosome with the recessive allele, small g means recessive allele. This is the X chromosome is received from the mother, always received from the mother. So, affected male get this allele from mothers. Next, females homozygous affected. Students, she is the female affected with the and also available with the homozygous recessive allele, homozygous affected, and all our sons are affected because of X chromosomes are always receive one of the X chromosome in sons receive from the female parent. Okay, So all our sons also affected. Another Y chromosome receives from the father. Next, affected females have affected father. Affected female, she is affected female, okay, and one of the X chromosomes receives from her father, okay. Affected females have affected father and carrier or affected mothers also, okay. Always receives from affected mother or also carrier. Next, X-linked recessive trait are passed on from affected father to 50% of his grandsons through its carrier daughter. Okay, students, which one is the inheritance gene from the parents to the offspring that causes the disorder? X-linked recessive disorder, students. X-linked recessive disorders, one of the gene that causes the phenotypic trait in offsprings. That is the gene inherited from the parents to the next generation. Which one is the gene that causes phenotypic trait that goes to the mutation? Okay, mutated gene is called recessive allele. Normal gene is called dominant allele. Okay, in this. Okay, that is the recessive gene, recessive allele inherited from the parents to the next generation and shows the abnormalities. Which are the offsprings that shows the recessive allele or the mutant gene causes different types of the abnormalities, phenotypic trait, okay, that is called disorders. X-linked recessive disorder, okay. X-linked recessive means normal gene that shows the phenotypic character goes to the mutation and changes into recessive allele, okay. That is the recessive allele is received from the parent and inherited from the parents to the next generation that causes the abnormalities that are called X-linked recessive disorder. Color blindness, hemophilia, Duchenne muscular dystrophy are the disorders. Example for X-linked recessive disorders. In this, first one is color blindness. What about the color blindness? It is X-linked recessive disorder, okay. And this is the color blindness firstly detected by Wilson in 1911. Okay, eye of the patient in color blindness, eye of the patients fail to distinguish red green color. Okay, student, in this retina of the eye in man contains cells sensitive to red green color. Phenotypic trait is genetically controlled. These students, color blindness, in color blindness disorder, retina of the eye shows the sensitivity to the red color and the green color and causes the disorder called color blindness, okay. And in this it is phenotypic trait is genetically controlled and its alleles are located on X chromosome, okay. That is the recessive allele is located on the X chromosome, okay. What about the inheritance pattern about the X-linked recessive disorder, that is color blindness? In these students, normal mother, 2 X chromosomes, these are having normal alleles, okay. Next, colorblind father. Colorblind is occurs due to the recessive allele present on loci of X chromosome. It is the Y chromosome. Plus means dominant and small g means recessive, okay. Normal mother marries colorblind father, okay. Colorblind male. Normal female marries with colorblind male. And if you observe, this is the parent generation. And if you observe F1 generation, F1 generated individuals are all are normal. But if you observe, females are carrier. females are carrier consist one of the X chromosome is with the recessive allele, carrier female, normal male. Okay students, and another, if married between the carrier female and the normal male for the F2 generation, if you observe 4 offsprings, in these 4 offsprings all are the females are normal, okay, all of the females are normal, and 50% of the males are male and another 50% are with the color blind. Okay student, this is the color blind father. Color blind father inherited is the recessive allele, okay, recessive allele to his grandson, students, to his grandson via the carrier daughter. Okay, it is the inherited pattern. Okay student, in this, women with normal vision, women with normal vision marries colorblind man, all sons and daughters are normal. Sons and daughters both are normal in condition, okay, but daughters are carriers. This is the daughter is with recessive allele with one of the X chromosome, but she is available with carrier female. Okay, when females show the type of the phenotypic character homozygous recessive allele, that means both of the X chromosome is with the small g. At that time only females shows the type of the color blindness. Okay, one of the X chromosome with dominant allele, another one is recessive allele. These are the females are act as carrier females. Okay, if a carrier woman, same type of the woman, carrier female, okay, marries man with normal vision, all daughters, these two are daughters, all daughters, half of the sons, in these two sons, half of the sons have normal vision and other half of sons with color blind. Okay students, 50% of the males with normal, another 50% of the males with color blindness. Okay, because color blind trait, this is the color blind trait, small g with X chromosome is inherited to his grandson with the help of the carrier daughter. Okay student, it is also called crisscross pattern of the inheritance. Students, now goes to discuss about hemophilia and Duchenne muscular dystrophy. These two types of the disorders also example for X-linked recessive inheritance. Okay, these are the disorders also shows the same pattern of the inheritance to the color blindness. Okay students, first what about the hemophilia? Hemophilia, it is also called bleeder's disease. Bleeder's disease, it is X-linked recessive disease. X-linked recessive disease, the mutant gene or the recessive allele is present on the loci of X chromosome, and by inheritance to the offspring from the parents, that causes the disorder, that is hemophilia, like a color blindness. In this, same pattern is available when compared with the color blindness, and one different is available in this hemophilia. That one is hemophilic female is do not survive. Okay, student, it is important. Female with hemophilia is not survive. Okay, and condition is lethal for females. Okay students, this is the condition, do not survive, survival capacity is losses. Female with hemophilia, okay, it is lethal for female, and it is caused due to the maybe cause of heavy blood. Most amount of the blood is losses at the time of the menstruation. Okay. Next, students, in patients of hemophilia, patients with hemophilia, blood is deficient of thromboplastin. It is thromboplastin is the important blood clotting factor. Student, thromboplastin is not formed in bleeders disease patients. Okay, this is the thromboplastin, formation of the thromboplastin. Thromboplastin is the one of the blood clotting factor. It is necessary to clot the blood. It activates the factor 7, okay, and motivate the formation of the prothrombin activator in the blood clotting mechanism. Okay, thromboplastin is not formed in patients with bleeders disease. Students, hemophilia is with two types. One is hemophilia A. Hemophilia A, it is characterized by lack of factor 8, anti-hemophilic factor A, that is the factor 8. Factor 8 is not produced in the blood, which it is plays a role in mechanism of blood clotting factor, clotting of blood clotting mechanism required for blood clotting. Factor 7 is lacks, it is not produces, which is required for blood clotting. Next, under hemophilia B. Hemophilia B, also called Christmas disease. What about that? Result from lack of plasmothromboplastine or factor 9. Factor 9, students, factor 9 also called plasmothromboplastine component. This is the factor is not produced in hemophilia B disorder and also called Christmas disease. Okay, person with hemophilia is injured bleeding, injured bleeding at the time of the injured bleeding is prolonged because clot is slow to form. Excess amount of the blood is losses through the injured region. Blood clot is slow to form. Okay, students. Next, under Duchenne muscular dystrophy. Duchenne muscular dystrophy. Duchenne muscular dystrophy, it is also one of the X-linked recessive disorder. In this X-linked recessive affecting around 1 in 3600 boys. Okay, students, and it is with progressive weakening of the muscles, loss of coordination. Students, what about Duchenne muscular dystrophy? One of the gene is responsible to code or formation of the dystrophin protein. Dystrophin is the protein present in the muscle tissue. It is helps to muscle fiber, strengthening of the muscle fiber, and it is the dystrophin protein connects the cytoskeleton filaments present in the cytoplasm to the extracellular matrix through the cell membrane or the plasma membrane or the sarcolemma. Extracellular matrix is connects with the cytoskeleton of the cytoplasm. It is possible by the one of the protein, that is dystrophin. Dystrophin is the protein coded by the one of the gene. That is the gene, student, that is the gene which codes the dystrophin protein that goes to the mutation and available in the form of the recessive allele. Okay, Dystrophin is the protein. It is not coded, not available in the muscle tissue, muscular tissue. That is causes the weakening of the muscles, progressive weakening of the muscles. Okay, students, in this it is with progressive weakening of the muscles, loss of coordination, and affected individual do not possess a dystrophin protein. Okay, disorder caused by a mutation in dystrophin gene. This is the gene, students, longest gene in the human beings. It is important. Dystrophin protein that coded by the one of the gene present in the karyotype of the individuals or the human beings. That is the gene is the longest gene among all the genes. Locates on Y chromosome. This is the gene locates on the Y chromosome, student, which codes dystrophin. Okay, dystrophin is protein connects the cytoskeleton of the muscle fiber to surrounding extracellular matrix through cell membrane. Connects sarcolemma and outer muscle filaments. Okay, supports matrix, students, outer muscle filaments supports muscle fiber strength. Okay, if the mother is the carrier for this Duchenne muscular dystrophy, if the mother is a carrier for this gene, half of our sons are affected with the Duchenne muscular dystrophy. Okay, another half of the sons and all of the daughters are not affected for this Duchenne muscular dystrophy. If the mother is carrier for this gene, half of our male children are expected to be affected. All female children born to carrier mother do not suffer from the Duchenne muscular dystrophy. X-linked dominant inheritance. That means X-linked, that is the responsible allele locates on X chromosome. Dominant inheritance, that is the gene is available in the form of the dominant allele that causes the genetical disorder in this type of the inheritance. Inheritance transmitted by dominant allele carried on X chromosome. Okay, allele responsible for genetical disorder locates on X chromosome. The responsible gene which causes the genetical disorder appeared on the dominant allele locates on the X chromosome. Okay, student. Next, one copy of allele is sufficient to cause the disorder. Student, X chromosome, it is the X chromosome. See, is X-linked because X chromosome. X chromosome is with the dominant allele. Okay, that is sufficient. Only one of the X chromosome is with the dominant allele. That is sufficient to cause the genetical disorder, to appear the phenotypic trait. That is the characteristic feature is possessed by the individual. Another one of the X chromosome is with the recessive allele, that the individual also shows the phenotypic trait. Okay, student. Next, all daughters of affected father will be affected. Okay, father, for example, father is affected, all the daughters also will be affected because of daughters always receives one of the X chromosome from the father. Okay, only one of the X chromosome is present in the father, that is with the dominant allele. This is the X chromosome is always enters into daughters from the father. So all the daughters also affected. All daughters of affected father will be affected. Students, if a mother is homozygous affected, that means both of the X chromosome is with the dominant allele, same position. X chromosome with dominant allele, another one is also with the dominant allele. Okay, next, all our children's, these are the X chromosomes enters into the her children's. So all our children's are also affected. Next, males are severely affected than heterozygous females. Heterozygous means one of the X chromosome is with the recessive, another one is dominant, that is heterozygous females. Males are severely affected than heterozygous female because males do not have normal copy of a gene to balance the effect of mutation on their simple X chromosome. Student, okay, males are mostly affected, severely affected, because than the heterozygous female. Males have only one of the X chromosome, that is with the dominant allele, and because in this position available in the males, these are the males are mostly affected, severely affected. Okay, there is no another one of the allele, opposite allele, and the another X chromosome. Males have only one of the X chromosome is with the dominant allele and severely affected than the females, heterozygous females. X-linked dominant trait includes, which are the example for X-linked dominant inheritance? These are follicular hyperkeratosis and incontinentia pigmenti. These two are very, very important, students. Okay, X-linked dominant traits. Example for X-linked dominant traits are follicular hyperkeratosis. Next, incontinentia pigmenti. In this first one, follicular hyperkeratosis. What is the meaning of follicular hyperkeratosis? Hyper means much, more. Keratosis, keratin protein. That means skin with excess development of the keratin in hair follicle. Okay, hair follicle that shows the excess amount of the keratin protein. Keratin is the one of the structural protein in the animals, in the organisms. Skin is with excess development of keratin in hair follicle. Okay, students, resulting in rough cone shaped elevated papillae. Students, okay, this is about the follicular hyperkeratosis. This is the symptomatic characteristic feature of the follicular hyperkeratosis, because it is the one of the genetical disorder occur due to the receiving of the X-linked dominant inheritance from the parents. Next, incontinentia pigmenti. What about incontinentia pigmenti? It is uncommon disorder, okay, and inherited in an X-linked dominant manner. Okay, next, it is lethal in some males. In some males, students, lethal, it is lethal in some males. Lethal means become mortal. Okay, become mortal, become into death. It is lethal in some males. Next, random loss. Okay, what about the characteristic feature of incontinentia pigmenti? Okay, random loss of melanin from skin leads to mosaic appearance of the skin. Melanin, it is the pigment secretes from melanocytes. Okay, it gives color to the skin of individuals. Okay, this is goes to random loss of melanin from the skin due to the receiving of the dominant allele from parents. Okay, random loss of melanin from skin leads to mosaic appearance of the skin. Next, students, a female IP, female incontinentia pigmenti, inherited the mutant allele from either of the parents. Okay, students, Next, homozygous incontinentia pigmenti in female is lethal. Homozygous incontinentia pigmenti, that means both of the X chromosome, that is the female having two X chromosomes, both of the X chromosomes is with the dominant allele about the incontinentia pigmenti. Okay, that is the female is lethal. Student, for example, it is discussion with the incontinentia pigmenti. Okay, female is normal, both of the X chromosome having the normal gene resemble with X small I, normal female, and incontinentia pigmenti male, X chromosome is with the dominant allele. Student, X-linked dominant inheritance, X chromosome is with capital I, dominant allele. Okay, that causes the phenotypic trait incontinentia pigmenti. Incontinentia pigmenti is the genetical disorder present in this individual. Okay, X capital I, Y. Okay, Y is normal. Next, gametes, X, both of the gametes are similar, X small I. Next, another sperm cells, these are the gametes of with X capital I, another one is with the Y. Okay, if you observe offsprings and the generator individuals, okay, in this females are affected. Okay, X with capital I, one of the X chromosome is with the dominant allele received from father, and another one of the X chromosome is with the normal gene. This is the capital I dominant allele or the mutant gene received from father. So female is affected with incontinentia pigmenti. Okay, that is the responsible gene, mutant gene or the allele received from the father. Okay, next another one, if you observe, male one is normal, X with the small I and the Y. Okay, normal individual, normal male. Okay, student, it is the inheritance pattern of the X-linked dominant inheritance with the example of incontinentia pigmenti. Linked inheritance. Y-linked means the responsible alleles or the genes present on the loci of Y chromosome. Okay, males only shows the Y chromosome in its karyotype. Student, called holondric inheritance. The chromosomes present on the genes present on the loci of Y chromosome, these are also called holondric genes. That is the chromosome, holondric chromosome. The inheritance pattern by this effect of these chromosomes, that is called holondric inheritance. Next, students, locates on non-homologous part of Y chromosome. Y chromosome is only one, that is present in the males. Females are the mother parent that does not shows the Y chromosome. Okay, these are the genes locates on the non-homologous part of Y chromosome. If a father is affected, if father will be affected, every son is affected, because Y chromosome inherited from the parent, from the father parent to his sons only. Female inherit X chromosome. X chromosome present in the father, that is inherited, always inherited to the female ones. Next, males hemizygous for these genes. These are the genes present only on the loci of Y chromosome. This is the single one in the males. So that is called hemizygous. Males are hemizygous for genes, for this type of the genes. Genes locates on the Y chromosome, only present in the males, and males are hemizygous for these genes. Next, SR5 gene present on the Y chromosome. This is the SRY gene present on the Y chromosome causes development for male characteristics. Male characteristics male characteristics that are shown by the individual, the responsible gene present on the loci of Y chromosome, that is SRY gene. SRY gene. Okay. Sex determining region of Y chromosome gene present on Y chromosome causes development of male characters. Next, student, which are the genes present on the Y chromosome that shows the Y-linked characters? Which are Y-linked characters? Which are the example for Y-linked inheritance? Hypertrichosis, students. Hypertrichosis is the best example. Hypertrichosis, that means excess growth of the hair on the pinna of ear. Pinna means outer ear, external ear. External ear that shows the excessive growth of the hair. Next, webbed toes. Webbed toes, it is also example. Web formation of the web between the toes. Webbing of toes. Next, student, and porcupine man. Porcupine man means stiff and strong hair present on the body surface. Body surface shows strong and the stiff hair, that is called porcupine man. Next, XY linked inheritance. Next one of the inheritance, XY linked inheritance. XY linked, the inherited genes present on the loci of X chromosome, Y chromosome. Okay, student, X chromosome, another one is Y chromosome. X chromosome, one of the X chromosome present in males and one of the Y chromosome present in males, and two X chromosomes present in the females. But these are the genes present on the loci of Y chromosome and X chromosome. Student, inheritance of trait by XY linked genes. XY linked genes. Genes locates on X chromosome, Y chromosome. These are causes inheritance of the one of the character which is shown by the individual. One of the trait, one of the phenomenon, one of the main character shows by the individual or the animal. The genes locates on X chromosome and Y chromosome. Student, incompletely sex linked genes. Student, these are the genes present on the chromosomes. Okay, and at the time of the division shows the crossing over. Okay, student, incompletely sex linked genes. Okay, next, and the genes present on pseudo autosomal regions of X and Y chromosome. Student, these are the XY linked genes, that are the genes causes for the appearance of the single trait, appearance of the trait, appearance of the character. Okay, and where these are the genes are present on X chromosome and Y chromosome, pseudo autosomal regions. Pseudo autosomal regions are the location in X and the Y chromosome. These are the regions shows this type of the genes, XY linked genes. Next, these are homozygous or heterozygous for traits. Student, for example, X chromosome shows that one of the gene, another one of the X chromosome, two X chromosomes present in the females. Both of the X chromosomes shows the genes. Okay, and locates at the same loci of chromosomes. That is called homozygous. Okay, student, and heterozygous. Heterozygous means, students, Y chromosome is with the one of the gene. Next, another, this is the X chromosome. This is the X chromosome, not a Y chromosome, because of presence of the only one of the Y chromosome that is with the one of the gene or the allele. Okay, that causes for the appearance of the heterozygous. These are homozygous or heterozygous for traits, for characters. Next, students, males genes on X chromosome inherit to females. Okay, X chromosome males. For example, if you observe male one, male is with the X chromosome with the allele. Okay, this is the allele is always inherited to females only. This is the X with the allele always inherited to the females, daughters. Next student, Y chromosome inherit to males. Y chromosome with allele always inherited into the YB plus inherited into the males or sons. Students, if you observe Drosophila females, in Drosophila females, XY linked alleles, X chromosome with alleles, Y chromosome with alleles. These are the alleles causes for the phenotypic trait, that is bobbed bristles. For bobbed bristles present on the same loci of X chromosome, Y linked alleles, X linked alleles. Two type of the alleles present on the loci of two X chromosomes present in the females. Next, males. Males students, alleles present on X chromosome and Y chromosome. Y chromosome is with allele, X chromosome is with alleles. Okay. Student, homologous regions of X chromosome, Y chromosome, that shows the two type of the alleles. Okay. Student, in this B plus is the normal allele. B plus is the normal allele. Causes normal hair, shows by the normal hair. Student, B plus is the normal allele, wild type. Okay. Next, B recessive. Student, it is the normal allele. B plus goes to the mutation. Okay, and appeared in form of the recessive allele or mutant allele, that is causes for the bobbed condition, bobbed bristles that shows by the Drosophila, due to the mutation of the B plus gene, B plus allele. Okay. Next, by the mutation of the B plus, another one of the recessive allele is formed, or mutant allele is formed, that is indicates with B. Okay, small letter B, causes bobbed bristles. Student, wild type male, X with B plus allele, Y is B plus allele. Next, bobbed bristles, it is the mutant condition. This is the gene goes to the mutation and causes recessive allele, XB, and another one is XB. Okay, student. And both of the alleles are same, only one indicates with only one in the zygote, XB. Next, two types of the gametes are produced, XB plus, YB plus. Okay, both of the gametes are united and forms the wild type female. Okay, students. And this is the YB plus is fused with the XB gamete, type of the gamete. Okay, and the same type, wild type female is also produces XB plus XB, wild type. Okay, normal hair. Next, and the students, in this XB YB plus, this it is also becomes into wild type male. It is the example for XY linked inheritance in Drosophila. The genetic disorders are divides into two types. One is Mendelian disorder, second one is chromosomal disorders. First one about the Mendelian disorders. What about the Mendelian disorders? Students, these are the disorders occur due to the single mutation in the DNA molecule. Okay, and these are the diseases also called monogenic diseases, because these are occur due to the changes in the sequence of the single DNA, single gene. Okay, a student, these are the disorders. Inheritance pattern is also observed, analyzed by the help of the pedigree analysis. Okay, students, these are the disorders also shows the pattern of the inheritance in family or group. Student, which are the Mendelian disorders? For example, hemophilia, sickle cell anemia, color blindness, phenylketonuria, thalassemia, Duchenne muscular dystrophy, albinism. These are the Mendelian disorders which shows the Mendelian pattern of the inheritance in the individuals, in animals. Students, first one about the hemophilia. What about the hemophilia? Hemophilia, that means inability to clot the blood at the time of the injury due to the deficiency of the clotting factor, blood clotting factors. Okay, hemophilia, it is divides into three types. One is hemophilia A, hemophilia B, second one hemophilia C. Students, hemophilia A, it is occurs due to the deficiency of the clotting factor 8. Hemophilia B, it is occurs due to the lack of the formation of clotting factor 9. Okay, student, and both of the hemophilia A, hemophilia B, also called X-linked recessive disorders. Students, these are the genes that regulates the formation of the protein factors, which is helpful in blood clotting mechanism. Students, clotting factor 8, clotting factor 9, these are inability to produce because of the which type of the DNA molecule that forms or synthesize the helpful to synthesize the blood clotting factor, that are the DNA molecule gene goes to the mutation. And in the observation it is appeared like a recessive allele. Okay, student, this is the recessive allele present on the loci of X chromosome. Okay, so these are the disorders also example for X-linked recessive disorders. X-linked recessive disorders, that means X chromosome is with the recessive allele. That is the recessive alleles, that is the gene goes to the mutation and appeared in the form of the recessive. That is, the recessive allele does not synthesize the protein factor, blood clotting factor, and causes the hemophilia A and hemophilia B. Student, these are the hemophilia A, hemophilia B also shows the crisscross pattern of the inheritance in population. Next, what about the hemophilia C? Hemophilia C, it is occurs due to the inability to form the blood clotting factor 11, blood clotting factor 11. Hemophilia B, it occurs due to the absence of the clotting factor 9. Student, hemophilia C, it is also called autosomal recessive disorder. Autosomal recessive disorder. This is the protein factor, that is plasma thromboplastin antecedent PTA, that is the blood clotting factor 11. It is synthesized by the DNA, that is the DNA locates on the loci of present on the loci of chromosome number 4, that is the autosome. Student, that is the DNA goes to the mutation and appeared in the form of the recessive allele. Okay, and that is now inability to synthesize form the blood clotting factor 11 causes the disorder. Hence it is autosomal recessive disorder, chromosome number 4, it is the autosome. So it is also called autosomal recessive disorder. Student, in this hemophilia disease, single protein that involves clotting of the blood is affected. Okay. Next, mostly it is occurs in females. It is the hemophilia is the disorder, one of the Mendelian disorder, genetic disorders mostly occurs in females because of 2X chromosomes present in the females. And students, 2X chromosomes present in the females. Both of the X chromosomes is with the recessive allele. That shows that are the individual shows the phenotypic effect or hemophilia disorders. Okay, and students, mostly occurs in females, and it is asymptomatic carriers of disorders. Student, mostly these are the individuals or the females asymptomatic carriers of disorders when one of the X chromosome is with the recessive allele, another one of the X chromosome is with the dominant allele. At that time, these are the individuals does not shows the effect of the hemophilia disorder, now called as asymptomatic carriers of disorder. Next, female double recessive. Females are compulsory 2X chromosomes with recessive allele, that is double recessive to express the hemophilia disease. Next, allele typically passed on from affected father to 15% of his grandson through his carrier daughters. Students, this is also called crisscross pattern of the inheritance. Hemophilia A by the lack of the clotting factor 8. Hemophilia B, that is by the lacking of the clotting factor 9. Hemophilia C by lacking of the clotting factor 11. Okay. Next, what about sickle cell anemia? Sickle cell anemia, it is the autosomal recessive disorders. The genes present on the loci of autosome, that is the autosome number is 11, chromosome 11. Okay students, RBC in this type of the disease, sickle cell anemia, in this type of the disease, red blood cells assume abnormal shape and rigid and sickle shape. Okay, gets sickle shape. RBC gets into sickle shape in hypoxia conditions. Hypoxia means low amount of the oxygen supplies to the red blood cells. Hypoxia conditions, these are the sickle shape red blood cells at the condition in sickle cell anemia. In this type of the disease, red blood cells are clump in blood vessels, gather, accumulate in the certain region of the blood vessels and causes physical weakness. Some amount of the symptoms, these are the symptoms of sickle cell anemia, and leads to physical weakness and pain and organ damage due to the low amount of the supply of red blood cells to the region of the organ. Okay, low supply of oxygen to that region, and paralysis also leads to paralysis. Paralysis means inability to move the muscles, lack the functioning of the muscles, that is paralysis. These are the symptoms of sickle cell anemia. A student, and this is the disease controlled by one pair alleles. This is the disease controlled by one pair of the alleles. One allele is HBA, another one is HBS. These two alleles present on the loci of chromosome 11. Chromosome 11 is the autosome. So it is the autosomal recessive disorder. That is the student, that is the allele is available in the form of the recessive condition. Which one is the chromosome that produces, that synthesizes the beta chain of the hemoglobin? Beta chain of the hemoglobin is synthesized by the one of the gene present on the loci of chromosome 11. That is, the gene goes to the mutation and changes into recessive form, that causes the disorder, that is sickle cell anemia. This students, chromosome 11 is with two alleles, one pair alleles, HBA, HBS. Student, homozygous individuals. Homozygous individuals, that means from the parents, these are the individuals receive HBS and HBS alleles. HBS, HBS, same alleles, both are same. So these are the individuals are homozygous. Homozygous individuals express the diseased phenotype. These are the individuals receive these type of the alleles. These are called homozygous individuals for sickle cell anemia. One of the allele from the male parent and another one of the allele from the female parent. This type of the individuals are homozygous individuals, compulsory to express the sickle cell anemia phenotype. Two alleles are sufficient to cause the sickle cell anemia. Okay, which individuals receive these type of the alleles, homozygous alleles from the parents? These are the individuals, homozygous individuals express the sickle cell anemia disease. Next, HBA, HBS. HBA, HBS. S indicates sickle cell and A indicates absence of that allele. Okay, HBA allele, HBS allele. Okay, student, both of the alleles receives from the parents. Okay, these are the individuals are with different alleles for sickle cell anemia. Okay, and called heterozygous individuals. Heterozygous individuals are unaffected for this disease. Sickle cell anemia disease is absent in these individuals, but one of the allele for sickle cell is present in these heterozygous individuals and act as a carriers for sickle cell anemia because of presence of the one recessive allele, okay, in his genotype. Shows healthy life. These are the heterozygous individuals with HBA, HBS, and shows the healthy life in these individuals. Next, student, caused by single mutation in DNA. It is the disease sickle cell anemia caused by single mutation in DNA. Okay, that is the DNA synthesizes or forms the beta globin chain of hemoglobin molecule. Hemoglobin molecule that is entirely formed with 4 polypeptide chains. In these 2 alpha chains and the one is 2 beta chains. In this, if you observe beta chain of the hemoglobin molecule, that is sequentially formed with the amino acids. Students, in the beta globin polypeptide chain, in the 6th position in the beta globin polypeptide chain, at the, if you observe 6th position, there is a glutamic acid. Okay, glutamic acid is there, and which one is the gene that synthesizes the beta globin polypeptide chain that goes to the mutation? By the formation of the recessive allele, that is the glutamic acid is not synthesized, and glutamic acid is at the 6th position. Okay, student, it is the 6th position is with the glutamic acid. When it is goes to the mutation, it is the glutamic acid is replaced by valine. Okay, student, valine. Okay, now it is forms the sickle cell anemia. It leads to the occurrence of the sickle cell anemia and chance to diseased phenotype. Heterozygous individuals, already you know heterozygous individuals shows resistance against the severe effects of the malaria. Heterozygous individuals, these are the individuals who shows the resistance against the severe effects of the malaria. Okay, these are the individuals also called heterozygous advantage about the sickle cell anemia. Phenylketonuria, it is discovered by a falling. Okay, Next, it is autosomal recessive metabolic disorder. Okay, one of the gene present on the chromosome 12, one of the gene present on the loci of chromosome 12 that goes to the mutation, that is the mutant gene that codes does not cause the synthesis of phenylalanine hydroxylase. Okay, it is the chromosome number 12, it is autosome. Okay, it is the autosome and is with the recessive allele, mutant gene that is also called recessive allele. Okay, autosomal recessive metabolic disorder. Student, which one of the gene present on the loci of chromosome 12 that produces, that synthesizes, that codes the one of the enzyme, that is phenylalanine hydroxylase enzyme. Phenylalanine hydroxylase enzyme converts phenylalanine into tyrosine. Okay, when this is the gene enters into the mutant form recessive allele, that one is the gene does not code the phenylalanine hydroxylase enzyme. Okay, and at the same time phenylalanine is by the absence of the phenylalanine hydroxylase enzyme, phenylalanine in the body converts into phenylpyruvate in the cells, cytoplasm of cell. It is the metabolic reaction. Phenylalanine is changes into phenylpyruvate. It is the phenylpyruvate accumulates in the body, accumulates in the tissues and causes the one type of the phenotype such as mental retardation mainly. That is about the phenylketonuria. Okay. Next, second one, thalassemia. What about thalassemia? Thalassemia, it is autosomal recessive blood disorder. Okay, autosomal, that one is the gene causes for the thalassemia, locates on the loci of chromosome, autosomal chromosome. So it is called autosome recessive blood disorder, and it is occurs due to the caused by excessive destruction of the red blood cell. Excessive destruction. Normally red blood cells goes to destruction after the lifespan is completed, 120 days is completed. Okay, but it is excessive destruction of the red blood cells due to abnormal formation of the hemoglobin molecules. RBC is with the hemoglobin chromoprotein, it is hemoglobin protein molecule. Hemoglobin, that is the molecule formed by 4 polypeptide chains. In these 2 alpha chains, 2 beta chains. Okay students, in this type of the disease, abnormal formation of the hemoglobin molecules. Okay, one type of the abnormalities occurs when hemoglobin is formed. Okay, by this type of the mutant gene in the thalassemia disease. Patients defect in alpha chain or beta chain. Okay, it is the thalassemia occurs due to the defects in the alpha chain, beta chain at the time of the formation of hemoglobin molecules. But in sickle cell anemia, it is occurs due to the changes in beta chain of hemoglobin molecule. Glutamic acid is replaced with valine. Okay. Next, it is thalassemia is a disease caused by production of the abnormal hemoglobin molecules. A student, in this type of the diseases, which type of the chain is affected? Based on this is the thalassemia is divided into 2 types: alpha thalassemia, beta thalassemia. Alpha thalassemia, student, in this type of the disease, production of the alpha chain is affected. Alpha chain is affected. Production of alpha chain is affected in alpha thalassemia. It is controlled by two genes. It is the disease controlled by two genes. One is HbA1, another one is HbA2. Okay, two types of the disease, two genes. It is the alpha thalassemia controlled by two genes. HbA1 is the one gene, another one is HbA2. Okay, these are the two genes present on the chromosome 16. Okay, chromosome number is also important. Next, caused due to mutation in one or four alleles. One gene with two alleles, already you know. In this, two genes are there. At the time of the two genes, which are the alleles are produces, formed, four alleles are there. Okay, one gene with two alleles, two genes with four alleles at the same time. Next, in this type of the disease, one type of the gene goes to a mutation, or any one of the gene, or one or four alleles are goes to the mutation. It is occurs due to the mutation in 1 to 4 alleles. Next, beta thalassemia. Beta thalassemia, it is also called Cooley's anemia. Next, in this type of the disease, it is also occurs due to the production of the beta globin chain is affected. Beta chain of the hemoglobin is affected at the time of the formation. A student, it is controlled by single gene. It is the disease is controlled by single gene, that is HBB on the chromosome 11. This type of the chromosome, in this it is the gene HBB locates on the chromosome 11. Next, alpha chains are more. In this type of the disease, alpha chains are morely produces, and these are the hemoglobin molecules or alpha chains are attached to the membrane of the red blood cell and also damage the membrane of the red blood cells, goes to the destruction. Next, color blindness. Color blindness, it is also called color vision deficiency. Okay, inability to see the particular colors. Okay, inability to identify the differentiation between the particular colors. Next, it is also called sex-linked recessive disorder. It is the color blindness is sex-linked. That means the type of the mutant gene present on the loci of sexual chromosomes. Okay, sex-linked recessive disorder. Next, student, due to the mutation of the gene locates on the X chromosome. Which one of this X chromosome? That is X chromosome. X chromosome is with the mutant gene to the type of the character. Okay, and it is occurs due to the mutation of the gene present on the loci of X chromosome. Next, student, this is the color blindness is differentiated into three types. One is protanopia. Protanopia, that is red color blindness. Inability to identify the red color. Red color blindness. Protanopia, red color blindness. Deuteranopia, green color blindness. Okay. Next, tritanopia, blue color blindness. In this, students, protanopia, deuteranopia, these are sex-linked recessive disorders. But in this tritanopia, if you observe, which is with the blue color blindness, it is autosomal recessive disorder present on the loci of autosome. Next, cystic fibrosis. What about the cystic fibrosis? It is the autosomal recessive disorder present on, that is the mutant gene present on the loci of autosome. Next, student, chromosome number is 7 due to the mutation of the gene on chromosome 7. Chromosome 7, it is the autosome in karyotype. Okay, that is the chromosome 7 is with the one of the gene, that is the gene is responsible for maintaining of the, helps in maintaining of the sodium chloride levels in the tissues, body tissues. Student, it is the gene goes to the mutation and causes the different types of the abnormal characters, and that leads to cystic fibrosis. Student, this is the gene maintains salt water movement across epithelial cell membrane. Okay, what is the function of this gene? This gene helps in maintenance of salt and the water movement across the epithelial cell membranes. Next, student, and by the mutant gene in the cystic fibrosis increases sodium chloride content in sweat. Sweat glands releases sweat. Okay, in amount of the sweat, sodium chloride content is increases. Next, increased loss of sodium water levels from respiratory epithelium. It is the another one of the symptom in cystic fibrosis. Mucus coats cells to become viscous and sticky. Okay, student, and mucus secretes by goblet cells. Mucus coats cells to become viscous, sticky, and mucus builds up in lungs and pancreas and gastrointestinal tract. Gastrointestinal tract, that means the inner surface of the food tube, and mucus also builds up in lungs and the pancreas. Chromosomal disorders. What about chromosomal disorders? These are another type of the genetic disorders. Second type. First one is Mendelian disorders caused due to the mutation in single DNA. Next, these are chromosomal disorders occurs due to the caused by abnormal number of chromosomes. There is a abnormality in chromosome number. Next, students, it is also occurs due to the error in equal distribution of the chromosomes at the time of the cell division into the daughter cells, mother cells to the daughter cells. Student, at the time of the division, there is a equal distribution of the chromosomes into the daughter cells from the mother cell. Okay, at that time, distribution of the chromosomes, there is a error. Next, it causes with the one is the cell is with increased number of the chromosomes, another one is the cell is with less amount the chromosomes. Aneuploid, that means it is the chromosomal aberration. Chromosomal aberration. Okay, that means gain or loss of one or more chromosomes in a set. Student, already you know about the diploidy and haploidy. Diploidy means double set chromosomes present in the nucleus, karyotype of the cell. That is diploid. Haploid means having the that is the cell having the single set chromosomes, that is called haploid. What about aneuploidy? Aneuploidy, that means one of the chromosome is less in number in total number of the chromosomes, or one extra chromosome is present in total number of the chromosomes. Okay, that is called aneuploidy. Gain or loss of one or more chromosomes in a set, in chromosome set of the cell, that is called aneuploidy. This is occurs due to non-disjunction of the chromosomes. There is a disturbance in distribution of the chromosomes from parent cell to daughter cells. That is causes that leads to aneuploidy situations. Next, chromosomes are two types. Chromosomes are two types. That is allosomes. Another one is next one is autosomes. Allosomes, also called sex chromosomes. These are XX in females, XY in males in human beings. First one, in allosomal disorders, Klinefelter syndrome. Klinefelter syndrome. It is caused by trisomy 23rd pair in males. Student, it is occurs in males, and it is occurs due to the trisomy 23rd pair. 23rd, which one is the 23rd pair? XX chromosomes in female, XY chromosomes in males. These are 23rd pair chromosomes. 23rd pair is allosomes, sex chromosomes. Okay, 23rd pair in male, trisomy. Students, instead of XY chromosomes present in the males, there is another one of the X chromosome is also present. Okay, that is the condition is called trisomy. Instead of one X, one Y chromosome in males, okay, and it is replaced with another one of the X chromosome, that leads to karyotype is with 47 total number of the chromosomes in karyotype. Total number of the chromosomes are 47, and it is by the another one of the X chromosome. Additionally, one of the X chromosome is present instead of XY chromosomes. 47 XXY is the karyotype. It is also termed with the trisomy 23rd pair in males. Student, what are the symptoms? Hypogonadism. Hypogonadism, okay, decreases the level in the development of the sexual or reproductive system organs. Next, reduced fertility. Fertility is reduces. Fertility is reduces in individuals, and feminine sexual development. Student, also these are the persons also shows the female sexual characteristic features. Next, not suppressed. Totally, this is the female sexual characters are not suppressed in individuals, and enlargement of the breast, and in these individuals somatic cells also exhibit bar bodies in their nuclei. Nuclei means number of the nucleus. Somatic cells exhibit bar bodies in nuclei. Next, student, Turner syndrome. What about the Turner syndrome? Turner syndrome, it is caused by monosomy 23rd pair. Student, karyotype is with 45. Total number of the chromosomes are 45 instead of 46 chromosomes. Okay, one chromosome is less. Okay, instead of one pair sex chromosomes, these are persons only with the single X chromosomes. Okay, that is called monosomy 23rd pair. 23rd pair is only X instead of XX chromosomes or XY chromosomes. One chromosome is lost, okay, in these individuals. Short stature and gonadal dysgenesis, okay, decreases the development of the reproductive system. Webbed neck, widely spaced nipples, and broad chest. Next, bar body is absent because of only one of the X chromosome is present in these females. Turner syndrome, this is the disorder occurs in females. Bar bodies are absent because of only one X chromosome. Next, student, autosomal disorders. What about the autosomal disorders? This first one is Down syndrome, second one is Edwards syndrome, third one Patau syndrome, fourth one Cray-Duchat syndrome, fifth one chronic myelogenous leukemia. Chronic myelogenous leukemia, also called chronic myeloid leukemia, also called chronic granulocytic leukemia. Student, in this first one, Down syndrome. What about the Down syndrome? It is occurs due to trisomy 21. Trisomy 21. Student, karyotype is 47 plus 21. Total amount of the chromosomes are 47 instead of 46. Plus 21. 21st autosome is with 3 in number instead of one pair autosomes. Student, 47 plus 21. 21st chromosomes are 3 in number instead of 2 or 1 pair chromosomes. And what are the symptoms? In Down syndrome, short stature and round head and furrowed tongue. Okay, student. Next, open mouth, partially open mouth, and physical, psychomotor, mental development is retarded. Okay, student. Physical development, psychomotor development, that means mental development, mental thinking. Okay. Next, mental development, totally these are the characters are retarded in these individuals. Next, student, Edwards syndrome. What about the Edwards syndrome? It is occurs by caused by trisomy 18. Trisomy 18. Karyotype is with 47 plus 18. Total number of the chromosomes are 47 instead of 46 chromosomes in human beings. Students, in this plus 18 means 18th chromosomes are 3 in number instead of 1 pair 18 chromosomes. Okay, 47 plus 18. One extra chromosome of 18th pair. Students, in this common in female offsprings. This is disorder or this syndrome is common in female offsprings. Next, mostly people die during fetal stage. Okay, which are the persons suffered with Edwards syndrome? These are the individuals are die at the time of the fetal stage. And infants also with a cardiac problem and kidney problem, cardiac kidney abnormalities also seen in these persons. Next, Patau syndrome. What about the Patau syndrome? It is caused by trisomy 13. Okay, students, karyotype is 47 plus 13. Total number of the chromosomes are 47 in nucleus and instead of 46. Okay, additionally one copy of autosome, 13th autosome. 13th autosome chromosomes are 3 in number. Hence the karyotype is with 47 plus 13. Next, students, these are the persons is with heart, kidney problems and brain, spinal cord abnormalities also seen in these individuals. Next, microphthalmia. Microphthalmia, that means less development of the eyes. Next, cleft palate. Cleft palate is also seen. Next, student, many infants die within the first days of the life. Okay, initial days of the life after the delivery from the mother. These are the infants goes to death. Next, students, Cray-Duchart syndrome. What about the Cray-Duchart syndrome? It is also called 5p minus syndrome. 5p minus syndrome. Cray-Duchart syndrome also called 5p minus syndrome. Okay. Next, Cat-Cray syndrome. A student, 5p monosomy. It is occurs due to 5p monosomy. What about the 5p monosomy? Students, in this partial loss of the 5th chromosome. Okay, students, 5p minus 5p monosomy. Karyotype is with 46 5p. Total number of the chromosomes are 46 in number as usual. Next, 5p minus partial loss of the 5th chromosome, that is autosome. The student, in this partial deletion of the short arm of chromosome 5. In this disorder, 5th chromosome, it is the autosome and it is at the time of the division. Okay, reaching of the chromosomes into the daughter cells. Partial loss of the short arm of the 5th chromosome that leads to the Cray-Duchart syndrome. Student, at the region of the fifth chromosome, that is with so small fifth chromosome in length, okay, that is so small. Next student, what are the symptoms appeared in cry to chart syndrome? Cat like cry of affected children's. Okay, these are the children's shows the cat like crying. Next, due to the problems, why these are the person shows the type of the character? It is due to the problems in larynx. Larynx is the sound producing organ in human beings. Due to the problems in larynx and nervous system, and it is also mentally, these are the persons also with mentally retarded. These are the children's usually die at the time of the childhood stage. Human Genome Project. First one, about the chromosome. Chromosome is a chain of DNA molecules wrapped in or coiled in eukaryotic cell. Next, student, nucleosome. Nucleosome, it is a linear series in DNA. Nucleosome, that is a linear series in DNA. One of the nucleosome that consists a group of the protein molecules. One of the nucleosome that is formed by 8 molecules of histone protein plus along with, that means 146 nucleotide pairs. One of the nucleosome that is formed by 8 molecules of the histone protein along with 146 nucleotide pairs, that forms the single nucleosome. Next, chromatin student. What about the chromatin? Chromatin, it is the DNA content along with the protein molecules in the nucleus of eukaryotic cell. Chromatin available in dense mass of loops and coils inside the nucleus that constitutes chromatin. Next, chromatin occurs in two forms student after the staining. One is a dark staining euchromatin, another one is a light staining euchromatin. Dark staining heterochromatin, light staining euchromatin. And another one, DNA that codes specific type of the proteins. These are the proteins involved in structure and the functioning of the living organisms. Next, some amount of the DNA helps in regulating the expression of the genes, and these are the genes that codes the specific type of the proteins, for example repressor protein. Remaining non-functional DNA, that is called junk DNA. It is that junk DNA does not code the any type of the proteins, not involved in synthesizing proteins. That is called junk DNA, non-functional DNA. Next, genome student. Genome, that means complete set of the DNA molecules in the organism. Complete set of the DNA molecules in living organism, that is called genome. Genome, it is the set of the DNA instruction in a cell. Next, DNA student. DNA, it is a chemical compound that contains genetic instructions need to develop the activities of the living organism. Next, student, genetic makeup lies in form of the DNA sequences in nucleus of the cell in living organisms. If two individuals differ, then their DNA sequences should also be different, at least in some places. Okay. Next, student, what about the Human Genome Project? Human Genome Project, it is the mega project, and international effort about the HGP, Human Genome Project, began in October 1990, and it is almost totally completed in by 2003. So it is a 13-year project. Okay, coordinated by US Department of Energy and National Institute of Health. Next, student, and HGP gives information knowledge about the DNA variations among the individuals and also helps in gives information in new way to diagnose, treat, prevent thousands of the disorders that affects the human beings. Next student, another HGP also associated with the rapid development of new era in human biology. Okay, it is also human genome project also helps in rapid development of human biology or bioinformatics. Student, also HGP also helps in providing clues to understanding human biology and learn about non-human being DNA sequences. Next student, also gives information in solving challenges in healthcare and also problems are there in agriculture. These are the problems are also solved by human genome project and energy production. Next student, genome of the bacteria and yeast and Caenorhabditis elegans. It is non-pathogenic nematode, Caenorhabditis elegans. Next, Drosophila plants like rice or Rhabdosis. These are living non-human being living organisms. DNA also have been sequenced by the help of the Human Genome Project. Students, silent features of Human Genome Project. In this, by the Human Genome Project, some amount of the observations drawn from Human Genome Project are as follows. In this, first one is human genome consists of chromosome. Human genome, the total the amount of the genetic material DNA molecules present in the nucleus of the eukaryotic cell of human beings that contain chromosome, which is made up of nucleotide base pairs that are present in the chromosome in human genome. Okay, next second one, average gene consists of 3000 bases. Average single gene that contains 3000 bases. Which are the bases? Adenine, guanine, cytosine, thymine. These are bases. One of the gene average contains 3000 bases, but sizes are different, with the largest known human gene being the one that codes for protein dystrophin. Dystrophin, it is the one of the protein present in the muscle fibers, helps in contraction and the relaxation of the muscle fiber. This is the dystrophin protein coded by the gene. Okay, that one is the gene is the largest gene in the human genome. Okay, dystrophin is the protein that coded by the one of the gene, that is the gene is the largest gene in entire human genome. Okay, it is the gene locates on the loci of X chromosome, short arm of X chromosome. It is present locates at the loci of short arm of X chromosome. Next student, and the third one, total number of the genes is estimated at 30,000. 30,000 genes present in the genome of human beings. Total number of the genes are 30,000. Next, all nucleotide bases are exactly same in all peoples. Okay, nucleotide bases are exactly similar in all peoples. Next, fourth one, functions are unknown for over 50% of the genes discovered. Next, fifth one, less than 2% of the genome codes for proteins. In total amount of the genome, less than 2% of the genome. Amount of the DNA content present in the genome, in this less than 2% of the genome codes for proteins. Next student, sixth one, repeated sequences make a very large portion in the human genome. Repeated sequences are stretches of the DNA sequences. These are that are repeated many times. Okay, in the DNA sequences, each one of the DNA that is made up with available in 2 strands. Okay student, in this DNA amount of the DNA that is with the sequences, nucleotide base sequences, and in this repeated sequences are also available in the stretch of the DNA sequences that are repeated many times. Next, chromosome 1 has the largest number of the genes. Chromosome 1 has the largest number of the genes. How many genes present chromosome 1? These are 2968 genes. 2968 genes present in chromosome 1. So it is the largest gene. Next, Y chromosome has fewest genes. It contains 231 genes only. Next, eighth one, scientists have identified about 1.4 million locations. 1.4 million locations where single base DNA differences occurs in human beings. Single base DNA differences, these are also called single nucleotide polymorphism, shortly pronounced as SNIPS. Single nucleotide polymorphism. Scientists have identified 1.4 million locations where single base DNA differences occurs in the human beings, genome of human beings. Student, these are the salient features of the human genome.